Hello, good afternoon students. I hope all of you are doing well and all of you have taken your exam, that is All India Rankers Test Series, in a very positive way, and I'm sure that all of you are going to score very good marks in the All India Rankers Test Series Minor Test 4. So with this, let us start with the paper discussion of AIRTS Minor Test 4. We are going to begin with zoology. So the date, class 12th, date is 28th of November 2022, class 12th and 12th plus. Subjects covered are physics, chemistry, botany and zoology. So this is a brief overview of the entire test that we are conducting. So number of tests are 4 minor, partial syllabus tests plus 2 mega, that is half syllabus tests, plus 6 full syllabus tests. The test pattern are same in line with the NEET exam pattern, that is 200 questions, 720 maximum marks, that is NEET pattern. Test duration is 3 hours and 20 minutes. Language covered is English. Alright, so let us begin with zoology with the first question. The good humor hypothesis was rejected following. So what was the good humor hypothesis? So first of all, you see from which chapter this question is. The question is from Human Health and Disease. And the question says, the good humor hypothesis was rejected following which of the following? Now, what is good humor hypothesis? See, it is mentioned in your NCERT that the ancient Greeks, including Hippocrates and the Indian Ayurveda system of medicine, believed or they apprehended about the good humor hypothesis, based on which they said that there are several bile which are present within the body. There are several humors which are present within the body, and of these, based on the concentration of a particular humor, the personality of the person depended. For example, people having black bile used to have hot personality and used to have fevers. Alright, but then following the discovery of the experimental discovery of blood circulation in humans by William Harvey and the discovery of normal temperature in people with black bile, the good humor hypothesis was summarily rejected. Alright, so first of all, the first option is invention of thermometer, discovery of blood circulation by William Harvey, discovery of anatomical position of the human heart, discovery of immunoglobulins. Now here you may get confused between the first option, that is invention of the thermometer, and discovery of blood circulation by William Harvey, because there is mention of thermometer along with blood circulation by William Harvey, but the thermometer was used to measure normal temperature in people with black bile. Alright, so it was not invented. So the correct option is discovery of blood circulation by William Harvey. So answer is B option. Let's move to the next question. The next question is asking you, sustained high fever, that is 39 to 40 degrees Celsius, stomach pain, weakness and constipation are the symptoms of which of the following diseases? First is pneumonia, typhoid, tuberculosis and cholera. Let us look at each of the diseases. Pneumonia. Pneumonia is a bacterial disease, right? Typhoid is also a bacterial disease. Tuberculosis is also a bacterial disease. Cholera is also a bacterial disease. But the correct answer over here is B, typhoid, right? Why? Because the primary symptoms or the most common symptoms of typhoid fever, which is also called as enteric fever, is sustained high fever, 39 to 40 degrees C, stomach pain, weakness, constipation, etc. Pneumonia primarily affects the lungs. Okay. There are two pathogens which cause pneumonia. One is Streptococcus pneumoniae and the other is Haemophilus influenzae. Typhoid is caused by Salmonella typhi. Tuberculosis is caused by Mycobacterium tuberculosis, and cholera is caused by Corynebacterium cholerae, right? So next is, which among the following is not a bacterial disease in humans? Plague, dysentery, common cold or tuberculosis. So we know that it is the common cold, which is a viral infection which is caused by rhinovirus. So this is not a bacterial infection. The correct answer is answer C. Next question. Filariasis is caused by a roundworm in which the parasite affects several organs but usually lives in the dash of human for many years. Right? So eyes, lymphatic vessels of the upper limbs, lymphatic vessels of the lower limbs or the liver. We know that the filariasis is caused by filarial worm. right? So the filarial worm lives in the lymphatic vessels of the lower limbs and thereby causes inflammation, that is edema of the lower limbs because of accumulation of the tissue fluid. It is the lymphatic vessels and the lymph nodes, they get blocked by accumulation of a number of filarial worms, right? So there are two filarial worms, one is Wuchereria bancrofti and the other is Wuchereria malai, as mentioned in your NCERT. So both of them, they tend to migrate to the lymph nodes and the lymphatic vessels of the lower limbs primarily. They also affect the genitals, causing gross deformities, but primarily they affect the lymphatic vessels of the lower limbs. They tend to block the pathways of the lymphatic vessels, thereby the tissue fluid will start oozing out. It starts getting accumulated in the lower limbs and causes its enormous massive inflammation, which makes the lower limbs look like that of an elephant. Hence it is also called as elephantiasis or filariasis. Let's move to the next question. Next question is PMNL, which is also called as polymorphonuclear leukocytes, and mucus coating form the dash and dash types of barriers in humans respectively, right? So we know that there are two different types of immunities in humans. One is innate immunity and the other is acquired immunity. Now when we talk about innate immunity, we have several barriers which have been mentioned in your NCERT: physical barriers, physiological barriers, cellular barriers, and cytokine barriers. So talking about the physical barriers, we have the skin, which also serves as a first line of defense. It does not permit the microbial pathogens to enter into the body. So skin is there, and then we also have the mucus coating of the epithelial lining of the respiratory tract, the gastrointestinal tract, and the urinogenital tract. So all of these form the physical barriers which do not permit the entry of pathogens into the body. The second type of barrier is physiological barrier. So physiological barrier is formed by the acid which is secreted by the cells which are present in the stomach, saliva in the mouth, in the buccal cavity, which comprises of an antibacterial enzyme named lysozyme. Okay, we also have lysozyme in the tear. So all these are physiological barriers. The third type of barrier is called as cellular barrier, in which we have different types of cells like PMNL, as mentioned, polymorphonuclear leukocytes or neutrophils. Okay, then we also have macrophage and we also have monocytes, right? So all these form the cellular barriers or the physiological barriers, right? So next question is, a patient suffers kidney failure and the doctor advises kidney transplant. So the patient has suffered kidney failure and the doctor has advised kidney transplant. After the transplant, the patient starts suffering high fever, diarrhea, vomiting, and other symptoms characteristic of an allergic reaction. Okay, so in allergic reaction, what happens, that there is a heightened response or an exaggerated immune response of the receiver or the patient towards a particular allergen. All right, so this is what has happened, that is the kidney has been recognized as an allergen by the patient. Which of the following components of immune system is primarily responsible for this situation? Antibody mediated immune response, humoral immune response, cell mediated immune response, or complement system? We know that it is a cell mediated immune response which is responsible for allergic reactions, right? So the correct answer here is cell mediated immune response. So we have two different types of immune responses within the body. One is antibody mediated immune response, which is also called as the AMI, which is also called as the humoral immunity, and the other is the cell mediated immunity, which is also called as the CMI. Okay, so while humoral immunity is mediated primarily by the B lymphocytes, the cell mediated immunity is mediated by 4 different types of T cells, right? Next, let's move to the next question. A 4-year-old child, while playing football in a grassland next to his house, accidentally steps on a huge cobra. Okay, so these reptiles, they tend to crawl around or creep around in these grasslands, okay, and get bit by it. So that child, the 4-year-old child, while playing football, gets bit by this cobra. He is rushed to a neighborhood government hospital where the doctor immediately administers a solution into his blood. Okay, now the question is, this injection which has been administered into the body fluid, that is the blood of the patient, contains what and it provides what type of immunity? So we know that there are 2 different types of immunity. One is active immunity and the other is passive immunity. So active immunity is a type of immunity which is elicited within the body after the exposure of antigen into it. So the antigen enters into the body and the body will recognize the antigen and will elicit a specific type of immune response in the form of production of antibodies or cell-mediated immune response. That is called as active immunity. So the body is actively eliciting a response. And what is passive immunity? When we directly administer the preformed antibodies into the body in order to help the body, or in order to supplement the body to ease its work to elicit an immune response. So the body does not have to be exposed to an antigen in order to show an immune response. It will directly be administered the antibody against a particular type of antigen. This is called as passive immunity. The body does not have to actively work, right? So the injection, that is anti-venom. Okay, so the injection will certainly contain antivenom. What is antivenom? Antivenom comprises of preformed antibodies, right? And what is the type of immunity? If it is a preformed antibody, it must be what type of immunity? It must be passive immunity. And again, passive immunity is again of two types. Just like active immunity is also of two types. It can either be natural or it can be artificial. Okay, so passive immunity is also of two types. It can be natural or it can be artificial. So let's talk about natural active immunity and artificial active immunity. So natural active immunity is when we are naturally, during the course of our lifetime, we are subjected to a particular pathogen, and then we elicit an immune response naturally. That is called as natural active immunity. What is artificial active immunity? When we deliberately introduce or administer a pathogen, either a complete pathogen which may either be weakened or attenuated or killed pathogen, okay, or we introduce a particular protein of that particular pathogen, so these, in order to elicit a type of immune response. So that is called as in the form of vaccines, right? So these are vaccines that we administer into the body. So this is called as artificial active immunity. Then we have passive immunity. What is passive immunity? Passive immunity, like we have already discussed, we directly administer the type of antibodies against a particular antigen. So we ease the work of the body to do it. So again, we have natural, passive immunity or artificial passive immunity. So the natural passive immunity is when crossing the transplacental barrier, we have IgG antibodies which are crossing from the mother into the fetal body. So that is one way in which we have natural passive immunity. Then we have artificial passive immunity, in which natural passive immunity has another example, that is colostrum, which is a golden yellow colored fluid which is secreted from the mother's mammary gland right after the birth, before the production of milk. So that has abundant IgA antibodies, right? So that is also an example of natural passive immunity. And the other examples of artificial passive immunity are anti-tetanus serum. For example, when we tend to fall down somewhere, we get a wound or a cut, then we are administered with ATS injection. So that also contains preformed antibodies, and it is not natural, so it is artificial. So artificial passive, one is ATS and the other is antivenom. Okay, let's move to the next question. One of the examples of vaccine produced from recombinant DNA technology is that against dash in dash. So it is a fact-based question. It has been very exclusively mentioned in your NCERT. So the correct answer is hepatitis B, and yeast, that is Saccharomyces cerevisiae, has been used as the host to produce the recombinant vaccine against hepatitis B. What is hepatitis B? Hepatitis B is a viral disease. Okay, let's move to the next question. Next question is, read the following statements and identify the incorrect option. Okay, so we have to identify the incorrect option. Biological response modifiers such as alpha interferons are administered in patients suffering from HIV. Let's move to the next option. Opioids are drugs that bind to specific opioid receptors present in our central neural system and primary reproductive structures. Next is macrophages serve as the HIV factory for proliferation of the virus and are destroyed with the release of new viral particles. So here we have this particular word that must be taken into consideration, that is destroyed. Okay, we must take care of the keywords which are creating an incorrect element in the statement. Okay, similarly here you see biological response modifiers such as alpha interferons are administered in patients suffering from HIV. Again, so if we are aware of why alpha interferons, which are biological response modifiers administered into the patient against which particular disorder and why, then we can easily encircle the key term. Okay, opioids are drugs that bind to specific opioid receptors present in our central neural system and primary reproductive structures. Again, we should encircle it. Next is lysozyme in tear and saliva is a physiological barrier of immunity in humans. This is a correct statement. So lysozyme is an antibacterial enzyme which is found in tear and saliva. Now let's see what's not so correct about the first three statements. The first statement is saying biological response modifiers such as alpha interferons are administered in patients suffering from HIV. It's not administered in patients suffering from HIV, it is rather administered in patients suffering from cancer. Alright, why? Because the neoplasm or the tumors which are so formed because of any type of cancer, they evade the immune response, right? So in order to help, in order to supplement the immune system to recognize these neoplastic tumors or the neoplasm, we introduce biological response modifiers such as alpha interferons, right? So this is incorrect statement. Next is opioids are drugs that bind to specific opioid receptors present in our central neural system and primary reproductive structures. Not primary reproductive structure, but the digestive tract. Okay, so this is also incorrect. Macrophages serve as HIV factory for proliferation of the virus and are destroyed. Macrophages are not destroyed, although they serve very much as the HIV factory, viral factories, but they're not destroyed after the virus is let out of the macrophage, right? So this is also incorrect. This happens to be the correct statement. So what is the correct answer? 1, 2, 3 are incorrect and 4 is correct, right? So what is the correct answer? Answer A. Let's move to the next question. Next question is, which of the following is not a secondary lymphoid organ, right? So very simple question. The correct answer is thymus, right? Because spleen, Pierce patches and tonsils are what? These are all secondary lymphoid organs, and thymus is a primary lymphoid organ, right? Let's move to the next question. Next question is what? Stanley Cohen and Herbert Boyer accomplished the synthesis of the first recombinant DNA by linking a gene encoding for antibiotic resistance with a native plasmid of. So this is related to the history of biotechnology, where we know that there were two people, namely Stanley Cohen and Herbert Boyer, who what did they do? They are credited with for formation of the first recombinant DNA, where what they did, they had taken out the antibiotic resistance gene and they had linked it with a native plasmid of which of the following? The native plasmid of Salmonella typhimurium or Salmonella typhi. Alright, so this is again a fact-based question, that is, they had used this particular strain of Salmonella typhimurium in order to extract the vector, that is the plasmid, the native plasmid, and then they took out the antibiotic resistance gene from some other organism, let's say E. coli, and then they took that isolated gene and then they incorporated or ligated that gene of interest into the native plasmid, which served as a vector and had been isolated from Salmonella typhimurium. Who did this? Stanley Cohen and Herbert Boyer. Right? Let's move to the next question. Next question is, the first restriction endonuclease to be isolated and characterized was which of the following? Again, it is a fact-based question, but I would like to give a little bit of historical overview of what really happened before this was discovered. So in the year 1963, okay, two enzymes were discovered in E. coli or in bacterium or in prokaryotes that were responsible to restrict the growth of bacteriophage. Now, what is a bacteriophage? A bacteriophage is a virus that infects bacterium, right? So the bacterium, as a part of its natural defense mechanism, it employs two different enzymes in order to restrict the growth of this bacteriophage. One of these enzymes was responsible to restrict the growth of bacteriophage by methylating it. So this was by addition of methyl group. This was called as methylation. Whereas the other enzyme was responsible to restrict in a different way, that is by cutting the DNA of the viral particles. So this cutting of DNA was called as restriction. Alright, and this enzyme, the second type of enzyme which was responsible to restrict the growth of bacteriophage by cutting its DNA was called as the restriction endonuclease. Right? So with this we had the discovery of the restriction endonucleases, and the first such restriction endonuclease to be isolated and characterized was HIND2. Now what is this? Why is it called as HIND2? It is called as HIND because H stands for the genus from which it has been isolated, namely Haemophilus right? And IN stands for influenzae, whereas the D and 2 refer to the strain, right? So this is how we can label the type of enzyme which has been isolated. Let's move to the next question. Eco R1 comes from Escherichia coli R1 RY10, Escherichia coli RY11, Escherichia coli RY12, Escherichia coli RY13. So here again it is a fact-based question. We must remember that Eco R1 comes from Escherichia coli RY13. Again, RY and 13 stands for the type of strain in which the numerical value refers to the serial number in which it has been characterized, right? Let's move to the next question. Identify the correct restriction sequence for Eco R1. Again, it is a question directly from your NCERT. So here we know what is Eco R1. It is a restriction endonuclease. So if it is a restriction endonuclease, it is going to cleave the DNA sequence at a particular recognition site. Now what is every restriction endonuclease has its own specific recognition site. We are well aware of that, right? And we have more than 900 different restriction endonucleases or restriction enzymes that have been isolated so far from over 230 different strains of bacteria. Each such restriction enzyme having its own recognition sequence, right? Now talking of the recognition sequence for Eco R1, which is also a restriction enzyme, which has been isolated from E. coli RY13, has its own specific recognition sequence. What is the recognition sequence? Recognition sequence, first of all, we know the answer is GAA-TTC CTT-AAG because it has to be complementary. Now let us take a look at how it really works. See, if it is going to cleave DNA, DNA is a double-stranded molecule, and both of the strands run anti-parallel to each other. That is, one will run from 5' to 3', the other will run from 3' to 5'. So let's say that this strand is running from 5' to 3', this strand is running from 3' to 5', right? So we have G, A, T, T, and C. Similarly, this has the type of nitrogenous bases which will be found on the complementary strand will also be complementary to the other strands. So with G, we know what is the base pairing rule. C, T, T, A, and G. That is, guanine will always base pair with cytosine, thymine will always base pair with adenine, right? And there is a triple hydrogen bond between guanine and cytosine and double hydrogen bond between adenine and thymine. Now the question is that how will Eco R1 come and recognize it? See, it will come, it is going to inspect throughout the length. See, this is not the only sequence or the part of the sequence which is found in the DNA. This DNA is a very long molecule, and the entire long molecule is a double-stranded DNA molecule containing several nitrogenous bases. But Eco R1 is going to come, it is going to inspect across the entire length of the DNA molecule, and then it is going to find out that where is this particular palindromic sequence found. It is 6 nitrogenous bases or 6 nucleotides long, right? And it has to be a palindrome. What is a palindrome? You read from beginning and from the end, it's going to read the same. That is called as a palindrome. For example, Malayalam, it is a palindrome, right? So here also we see that it is a palindrome, read from left to right and read from right to left. So this is a palindrome, it is 6 nucleotides long. So Eco R1 will come, it will inspect the entire length, it will find that yes, this is the type of recognition sequence that I'm supposed to come and work on. Now how will it work? It will come, it is going to cleave the DNA at this side between G and A and at this side between G and A. Okay, so resultantly what will we get? As a result of this cleaving, what we obtain is this. Right? So one of the strands that we will resultantly obtain will be this, that is 1G will be here, 5', and 3'. So C, T, T, A, and A. So this will be one part that we'll get. The other part that we'll obtain will be this. That is this part, right? So here we have what is left over, A, T, T, C, 3', right? And here we simply have this, that is G, and 5'. So this is what we obtain as a result of cleaving of eco R1, right? So this is what the question is asking you, and the correct answer is C. Let's move to the next question quickly. The fragments of DNA obtained by restriction digestion can be separated by the technique of. So we know that whenever we conduct a recombinant and rDNA technology technique, so in that there are several steps starting from identification of the source, from which source we need to get the DNA, the gene in question. So after identifying the source, we get the DNA. Okay, now that DNA, we cannot directly pick up the gene and isolate it from the organism. We do not have a technique as of yet for that. So what we have to do is we have to get the entire genome out. Okay, so that is called as genome isolation. Or we have to get the entire DNA pool out of the nucleus of the cell. So first of all, identification of the source. Second is the isolation of the DNA. Then third is fragmentation of this DNA in order to form different pieces of the entire or the whole DNA, so that we can get that fragment in which the gene in question is present, right? The third step is identification of the desired fragment from that pool of fragments, right? The next is separation of that particular fragment. Fourth, the next step is in order to ligate that particular desired fragment of DNA with a vector to form a recombinant DNA. The next step is to incorporate that recombinant DNA into a host, and finally we get the host to produce or synthesize the product which is desired. So these are the different series of steps. Now here they are asking you the fragments of DNA obtained by restriction digestion. So this is referring to which particular step? This is referring to the second step, in which after having isolated the entire DNA, we have cut it into different fragments using restriction enzymes, right? Now the next step was what? The next step was to separate those fragmented DNA strands. So the technique which is employed to separate the fragmented DNA strands is called as gel electrophoresis. Now what is gel electrophoresis? In gel electrophoresis, we separate the fragmented DNA strands based on their molecular size. Okay, so the smaller the size, the farther it will migrate. Now what is the principle based on which we operate gel electrophoresis? It is based on the application of electric field. So what we do, we keep the DNA fragments in different wells, we apply an electric field, and we know that one of the electrodes will serve as a cathode, the other electrode will serve as an anode. What is the cathode? Is it positively charged electrode or negatively charged electrode? Cathode is the negatively charged electrode. What is anode? Anode is the positively charged electrode, right? So DNA, being a negatively charged molecules, upon application of an electric field is going to migrate from the cathode towards the anode. Okay, so as and when it is migrating by the sieving effect which is produced due to the matrix of the agarose gel, the smaller the fragment, the farther it is going to migrate. So using this technique, which is called as gel electrophoresis technique, we separate the different restricted fragments. So the correct answer here is agarose gel electrophoresis. What is agarose? Agarose gel is a polymer which is a natural polymer which is obtained from seaweed, right? Let's move to the next question quickly. Next question is, as an important step of recombinant DNA technology, the host is made competent by treating it with. See, so amongst the different steps that we had discussed of the recombinant DNA technology, one step was to introduce, or maybe the last but one step was to introduce the recombinant DNA or the recombinant plasmid into the host, so that the host can express the foreign gene that has been incorporated, or the gene of interest which has been incorporated. So when we generally use the hosts which may be microbial cells, so these microbial cells do not actively take up or proactively take up the DNA or the recombinant DNA. So we have to make the host competent to take up the recombinant DNA. So there is a particular way by which we do it. We treat the host with a specific concentration of divalent cation, and we also subject it to heat shock treatment. So what is heat shock treatment? Heat shock treatment is, first of all, we pour a specific concentration of divalent cation on the host. Then the plasmid will come and it will settle down on the outer surface of the host, let's say the cell wall of a bacterial cell. Then we give heat shock treatment, in which we are generally keeping this entire apparatus on an ice bath. So we suddenly keep it, transfer this entire setup to, let's say, 42 degrees C, and all of a sudden we take it back to ice bath. So this is called as heat shock treatment, following which the DNA which had precipitated on the surface or the cell wall of the bacterium will be taken up into the cytosol of the bacterium. Alright, so this is called as heat shock treatment, but here what is required is specific concentration of a divalent cation like calcium 2+ ion. So let's move to the next question. Next question is, biolistics or gene gun method suitable for plant cells uses, right? So biolistics or gene gun method which is suitable for plant cell, what does it do? Again, it is a fact-based question amongst the different ways by which we introduce the foreign DNA into the host cell. Biolistics is one way. For example, microinjection. Microinjection is suitable for animal cells. Biolistics is suitable for plant cells. Then we also have disarmed pathogens. So there are different ways in which we can introduce the foreign DNA into the host. So here, biolistics, what we do, high velocity gold or tungsten particles which are coated with DNA, they are bombarded on the host cell, which in this case is the plant cell, right? So here the correct answer is high velocity gold or tungsten particle. Let's move to the next question quickly. Which of the following is used for precipitation of DNA? So we know precipitation of DNA. Again, the first step is isolation of DNA. So in isolation of DNA, what we do, if we have to isolate DNA from any particular source, it can be either a bacterial source, that is a bacterial cell, or it can be a plant cell, or it can be an animal cell, it can be a yeast cell. So depending on the type of cell that we are using, as that we have identified as a source, we have to use our technique. For example, if we have identified a plant cell as a source for DNA, so what we have to use that enzyme which is going to digest the cellulose containing cell wall, that is cellulase enzyme. Because we have to disrupt the cell, where is this DNA present? If we have to isolate DNA from the source, where is the DNA found? It is found in the nucleus. Nucleus is in the cytoplasm of the cell. So to reach the nucleus, we have to first disrupt the outermost covering, which is what? Cell wall in case of plant cell, cell membrane in case of animal cell, again cell wall in case of yeast cell, again cell wall in case of bacterial cell, although the composition of the cell wall is different. So again, depending on what is the outermost layer, we have to use the isolation technique. For example, if we have to use, let's suppose, a plant cell, so for that we have to use cellulase enzyme to disrupt it. Okay, so if we have a bacterial cell, so we have lysozyme enzyme to disrupt the the cell wall of bacterium. If we have an animal cell, we do not require much, right, because it does not have a cell wall at all. If we have a yeast cell or any fungal cell for that matter, we will have to use chitinase enzyme, because chitin is what forms the cell wall of this fungal cell. So on and so forth. So what we do, we disrupt the cell, and then whenever we get the DNA, in order to restrict the DNA or in order to digest the DNA, when we conduct restriction digestion for the DNA that we have so obtained, it has to be made sure that the DNA is in pure form. But whatever macromolecule we have got out of breaking or disrupting the cell, it does not necessarily contain DNA in its pure form. Why? Because it may contain protein also, it may contain carbohydrate also, lipids also, RNA also, so on and so forth. So in order to proceed with the restriction digestion, we have to first purify the DNA. So for purification of the DNA, what we need to do is, we have to use the enzymes which are going to digest the macromolecules which are apart from DNA. For example, if we have to digest the RNA, then we have to use RNase enzyme. If we have to digest protein, we have to use protease enzyme. If we have to digest the lipid, so we have to use lipase enzyme. Why? Because it is not the DNA which is only coming out. Along with DNA, a number of other types of macromolecules are also coming out, right? So we have to use all those enzymes for purification. And after purification, we have to precipitate the DNA. So for precipitation of DNA, we use chilled ethanol. Okay, so the moment you trickle down chilled ethanol dropwise, you will find that DNA has started to precipitate in the form of white, fine thread-like structures. It's a beautiful view, right? So, and then you finally spool it out using an apparatus. So what do we use here? It's chilled ethanol that we use, right? Let's move to the next question. Next question is what? Agarose gel electrophoresis is used to check the progression of restriction enzyme digestion. DNA moves from where to where because it is dash charged. So just now we have discussed what is gel electrophoresis. In gel electrophoresis, we use that apparatus in which we use agarose gel, which is a natural polymer isolated from seaweed, and then we subject the DNA, which is a negatively charged molecule, to an electric field, because of which by the sieving effect, DNA is going to move or migrate from cathode towards the anode. Why? Because anode is the positively charged electrode and cathode is a negatively charged electrode, right? So, and smaller the size or smaller the fragment of DNA, the farther it migrates. So what is the correct answer to this question? It moves from cathode to anode, that is negatively charged respectively, right? It is a very simple question. It was a bonus question maybe for all of you. Next question, which of the following properties of plasmid cannot be taken as a selectable marker? Now, what is a selectable marker? We know that some synthetic plasmids like PBr322 and PUC8 have been used as vectors for selection of recombinants or for differentiating recombinant from non-recombinant, and antibiotic resistance genes more often than not have been used as selectable markers. So you can use any type of, when you are, since it's a synthetic plasmid, so you can design the plasmid based on how to meet the requirements of differentiating the recombinant from the non-recombinant. So if you are designing an experiment, in that you can use any type of antibiotic resistance gene as a selectable marker, be it ampicillin resistance, or it can be tetracycline resistance, chloramphenicol resistance, kanamycin resistance, any antibiotic you can use, and depending on the type of antibiotic resistance gene that you have used as a selectable marker, you can design yours the further steps of the experiment in order to differentiate. So in PBr322, we use two different types of antibiotic resistance genes, namely, one is ampicillin resistance gene and the other is tetracycline resistance gene, right? So I think I also have a picture of that PBr322 here. Yeah, so this is one picture in which we have two different antibiotic resistance gene. One is the ampicillin resistance and the other is tetracycline resistance, right? So this is how we design. Let's move back to that question. So which of the following properties of plasmid cannot be taken? So ampicillin is an antibiotic resistance, tetracycline, chloramphenicol, origin of replication of course cannot be taken as, so this is the correct answer to this question. Let's move to the next question. Identify the option that does not form the component of a bioreactor. Now a bioreactor, if you try to recall what is the structure of a bioreactor, what are the different parts which are found in a typical bioreactor, you will see that this is the structure that we see. Okay, so what all do we find here? Flat bed impeller is there, flat bladed impeller is there, inlet for steam is there, inlet for steam is there, right? So you see here we have an inlet for steam over here. Okay, steam for sterilization, this is the inlet for steam. Then foam breaker is also there. You see here foam breaker is found, right? And then we have inlet for waste like ammonia. So all these things are found, inlet for waste like ammonia. So we have all these things, but D, that is inlet for waste like ammonia, so this is not found, right? So what is the correct answer? Inlet for waste like ammonia. That does not form a component of a bioreactor. The rest, all three, they form a component of the bioreactor. Let's move to the next question. Next question is, microinjection does not form an ideal choice as a method for gene transfer for which of the following cells? So we have seen that whenever we are introducing a foreign gene into a host, then depending on the type of the host, we select our method. Okay, so here hepatocyte is what? It is an animal cell, so microinjection can be used. Then yeast, it is a unicellular fungus. So here maybe it cannot be used, but let's take a look at the other options first. Next is WBC, it is an animal cell, microinjection can be used. Leukocytes are nothing but WBCs, so animals, microinjection can be used. Yeast should be the correct answer here, because more likely we are going to use biolistics for this particular method, for this particular host. Let's move to the next question. Identify the restriction enzymes that are used to digest the site of ampicillin resistance in PBr322. Again, it is a fact-based question. You must recall that whenever we try to recall the structure of PBr322, we see that it is PVU1 and PST1 which are the restriction enzymes which are used to cleave the site of ampicillin resistance. So the correct answer here is PVU1 and PST1. We can also see over here that this is the site of ampicillin resistance gene which can be cleaved by two different restriction enzymes, as has been categorically mentioned by your NCERT. You must keep it in mind. All the figures are very important. That is PVU1 and PST1. Right? Let's move to the next question. An ideal plasmid vector should have dash cloning sites and dash copy number. Choose the option that fill the respective blanks correctly. Right? So first of all, we know that any ideal vector preferably should have one cloning site. Okay, the lesser the better. Why? Because the more the number of cloning sites, more the number of fragments are generated, it complicates the process, both of isolation of DNA and that of formation of recombinant DNA, right? So it should have minimum or regulated, okay, that is an ideal plasmid vector should have dash cloning sites and dash copy number. So it should have regulated cloning sites and low copy number. All right. So the correct answer here should be C and not A. So we're going to change this. Let's move to the next question. PVU2 is a restriction enzyme used at which of the site of PBr322 plasmid? One is tetracycline resistance, ampicillin resistance, ROP and ORI site. Again, you see here it is a fact-based question. The moment we get such type of questions, we must recall what is the structure of PBr322 and at what site what particular restriction enzyme is indicated, right? So here we see that it is the ROP site, that is repressor of promoter site, which can be PVU2 is used as this at this ROP site. All right, let's move to the next question. Next question is Bt toxin gene has been cloned from. So it has been cloned from where? It has been cloned from bacterium. And what is the name of that bacterium? It is Bacillus thuringiensis. Right. So this is the bacterium from which Bt toxin gene has been isolated. Let's move to the next question. Which part of the tobacco plant does Meloidegain incognita infect, thereby significantly reducing? So we know that it is a nematode which infects the root plants of tobacco. That is the root of tobacco plant, right? So the correct answer here is root of tobacco plant. Very easy question, very simple question. Let's move to the next question. RNAi is initiated by. Again, if we know what is RNA interference or if we know what is gene subtraction or gene silencing, so here what do? It is the double-stranded RNA which is formed. How does this process work? This process works by silencing the RNA which is transcribed from, that is the pathogenic RNA which is transcribed from the pathogenic DNA of the host cell. Okay, now what do? We insert a double-stranded DNA that is within the tobacco plant, in which both sense and antisense, we insert a gene within the tobacco plant which will produce both sense and antisense strands of DNA, right? So those sense and antisense strands of DNA are going to transcribe themselves to form a double-stranded RNA. So this double-stranded RNA or a dimer of RNA, which forms very much a part of the tobacco plant, when this nematode is going to feed on this particular tobacco plant, then this double-stranded RNA will go and it will hybridize with the single-stranded RNA of the nematode, thereby silencing the pathogenic activity of that particular RNA. Right? So this is how it functions. Here the correct answer is double-stranded RNA. Let's move to the next question. Which of the following organizations has the Indian government set up to make decisions regarding the validity of GM research? What is GM research? Genetically modified research and safety of introducing genetically modified organisms for public services. So again, it is GEAC. It is very clear to all of us, and here Genetic Engineering Approval Committee is the abbreviation for that particular body, government body, right? Let's move to the next question. In 1997, the first transgenic cow, Rosie, produced human protein-enriched milk with a concentration of, it is very much a fact-based question from the chapter Animal Husbandry, so there is nothing new over here. The correct answer is 2.4 gram per liter, and there was also a protein which was enriched in this milk which was called as alpha-lactalbumin protein, right? So next is, for testing chemical safety or toxicity of drugs, transgenic animals are made dash sensitive to drugs. They are made more sensitive to drugs. Why? There is a reason behind it, because it's going to save time, right? So how is it going to save time? Ideally, if it was sensitive to a particular drug, then the amount of time that the non-transgenic animal would take to elicit an immune response to a particular allergen or to a particular antigen would be some amount of time. But if that organism or the host is made more sensitive, or its sensitivity to a particular allergen or antigen is heightened, okay, or it is increased, then in a small concentration of, or introduction of a small concentration of that particular antigen or allergen will be sufficient to elicit an immune response, right? So we have reduced the time required to see the effect or to study or to monitor the physiological effect of a particular antigen or the sensitivity of a particular antigen to a particular host, right? So here it should be made more sensitive, right? Let's move to the next question. The biggest advantage of using transgenic animals for chemical safety testing of drugs is that the process can be done in less time. Again, it's a related question, so this also turns out to be a bonus question for all of you. Let's move to the next question. Milk produced by rDNA technology as a biological product was nutritionally rich in, again, alpha lactalbumin, 2.4 grams per liter, rosy cow. All right, so all these things are very much related. The moment we get such questions, we tend to recollect all those facts together. All right, let's move to the next question. Next question is what? Which of the following animals are used for testing the safety of polio vaccine? Again, very specifically mentioned in your NCERT. It is the transgenic mice which have been used for chemical safety testing of polio vaccine, right? Let's move to the next question quickly. The first clinical gene therapy was, so next we move to section B, in which the first question is, the first clinical gene therapy was given in 1990 to a 4-year-old for deficiency of which of the following enzymes? We are very well aware that the type of enzyme which was deficient in that 4-year-old was ADA. Okay, so this enzyme is required for maturation of lymphocyte, and hence it is adenosine deaminase, which is the correct answer. All right, let's move to the next question. The most probable permanent cure for ADA deficiency could be insertion of relevant gene into cells at an early embryonic stage. Why? Because if we just change the WBCs, let's suppose that we are just injecting the WBCs, or we are, first of all, we are isolating the WBC, we are culturing the WBC in under lab conditions, and then we are introducing a proper WBC for its maturation, and then what we do, we take that WBC out of the lab conditions and introduce it into the patient. But again, if we do it, WBC lifespan is not very much, right? So we will have to repeatedly do it again and again. So that is one challenge. Another challenge is when we conduct, let's suppose, enzyme replacement therapy, again that enzyme is not going to be retained or be functional for a very long period of time. So we will have to repeatedly reintroduce enzyme again and again. So the only permanent cure that is in sight is to introduce the cultured cells at an early embryonic stage, right? So this is the most probable permanent cure. Let's move to the next question. The intracellular process for RNA interference is found in. Okay, so we know that RNAi process or RNA interference process or gene silencing process is found in all eukaryotes as a natural defense mechanism. Okay, so here the correct answer is all the eukaryotes. Next question. The Bt cotton produced used the natural phenomenon of certain dash that produced some protein crystals during a particular phase of their growth. These protein crystals contained inactive insecticidal protoxins that were activated in the gut of the insects due to exposure to which of the following, right? So bacteria and alkaline. So of course it has to be bacteria, because what is the source of that gene? The source of that gene is bacterium. What is the name of the bacterium? It is Bacillus thuringiensis. So what is the principle behind this entire process? We see here that some of the insects, they feed on Bacillus thuringiensis bacterium. Those insects that are responsible to destroy, let's suppose a locust or any other pest for that matter. So let's suppose that that particular pest is supposed to destroy a cotton crop, and that as a part of its feeding habit or as a part of its food is ingesting on Bacillus thuringiensis also. So we learnt somehow that the natural way by which this Bacillus thuringiensis protects itself from its predator, that is the pest in this case, is by producing some crystals. So those crystals contain some insecticidal proteins. Now the question is, why are these crystals not destroying the bacteria if it is produced within the bacteria? Because they're found as protoxin or in an inactive state. How will they get activated? Interestingly and surprisingly, in a genius a grand scheme of universal design, you see that this particular protoxin remains as a protoxin, thereby not killing the bacterium when it is not ingested by the predator. But once it gets ingested by the predator, it's going to reach the gut. So the moment it reaches the gut of the insect, so it gets activated, right? So it gets activated due to exposure of what? Alkaline pH. Right, so in the gut of the insect there is alkaline pH, there it will get activated, and when it gets activated, the protoxin gets converted or activated into toxin, active toxin. This active toxin is going to go and bind itself with the epithelial cells of the gut, they're going to create pores over there, water will enter, it will cause lysis, and thereby the insect is going to starve to death. So you see that this is the mechanism that we have learnt from nature, that is, we have learnt from Bacillus thuringiensis and have applied back to nature in order to protect nature itself, right? So what we have done is we have learnt this particular process, and then we have applied it by introducing the same gene in tobacco plant, so that whenever this pest feeds on tobacco plant, it's going to die and the tobacco plant will be saved, right? So this is how we have done it. Next is, so the correct answer over here is bacterium and alkaline condition of the pH of the gut of the insect. Let's move to the next question. The primary advantage of PCR is that it enables us to detect the presence of pathogens at dash concentrations, because it can produce dash copies in dash cycles, right? So again, it is a fact-based question. We require very low concentration of the source DNA in order to amplify it, right? So 1 billion copies in about 20 cycles. And what is the formula? The formula is 2 to the power n. We are well aware of that. So let's suppose that if we have 30 cycles, that this goes to approximately 1 billion copies, right? So this is how we get to know about it. Let's move to the next question. The milk yield of cattle is primarily dependent on quality of breeds in the farm. Very, very simple question, directly from the NCERT, right? A simple statement that has been taken as it is from the NCERT, that is primarily dependent. Now you may argue that, sir, why not fodder, why not cleanliness and hygiene? Of course, on these factors also, but primarily dependent upon quality of breeds in the farm, because if the quality of breed is not good, no matter how clean you keep the ambience, no matter how good the fodder is, and no matter how skilled the milkman is, you're not going to get good quality milk, right? So the correct answer over here is C. Let's move to the next question. A group of animals related by descent and similar in most characteristics directly from your NCERT, it is a breed. Okay, so that is, they're common in their features, size, configurations, everything. It's called as a breed. Let's move to the next question. Next question is, cross between animals of different breeds is called as. What is it called as? It's called as cross breeding. Different breeds. So we know that inbreeding is something that seeps in because of, that is, inbreeding depression is something that seeps in because of continuous inbreeding. What is inbreeding? Inbreeding is the breeding or mating between more closely related individuals of the same breed, right? So that is called as inbreeding. So because of continuous inbreeding, we have homozygosity that seeps in, because of which we also have inbreeding depression, right? So in order to tide over this inbreeding depression challenge, we conduct two different processes. One is the outcrossing and the other is crossbreeding. So what do we do in outcrossing? And both of these taken together is called as outbreeding. So what we essentially do in outcrossing is that we take two different individuals of the same breed that are not related on either side of the pedigree for at least 4 to 6 generations, right? So when we take those two different individuals and we conduct a cross between them, it's called as outcrossing, and the progeny so formed is called as an outcross. Similarly, we do crossbreeding. In crossbreeding, it is very simple. We take high yielding variety male and high yielding variety female, that is superior quality male and superior quality female from two different breeds, and then we conduct a cross between them. So it is possible to get a fertile offspring, of course, because although they are from different breeds, but they are from the same species, right? So there's no problem. We can have a high yielding variety of progeny in case of crossbreeding also. So outcrossing, crossbreeding, both of them together are called as outbreeding, right? So this is what we do here. So if two different breeds, automatically it is crossbreeding. All right, let's move to the next question. Next question is what? Inbreeding is the process of identifying superior males, superior females, and mating them to produce offspring and continuing the process for how many generations? Again, it is a fact-based question. We should just remember that it is for 4 to 6 generations. That is all. Right? Let's move to the next question. Next question is what? The advantage of inbreeding is the development of dash lines, pure lines, or what Mendel had called true breeding varieties, right? So when we try to recall the experiments of Mendel, so he had selected true breeding varieties, although he didn't know that they were true breeding. He was lucky in that case that he got homozygous parents only, but then they were homozygous parents. So anyway, they're called as true breeding. Why do we have to have true breeding? Because we wish to retain the desirable characteristics of the organisms that we see. So if we suppose that there's high milk yield in a particular female and there's high musculature in a particular bull, in a particular male, right? So in that case, we wish to retain these two good qualities because random matings can dilute the qualities over generations. So for that, inbreeding is carried out in order to retain. But the problem is that since it's not it is not a gene specific hybridization, it's a general hybridization. So by default we will have some undesirable genes also seeping into the next generation along with these desirable characteristics, right? So selective breeding comes to our rescue in that case. So here the advantage of inbreeding is the development of pure lines, good enough, but the disadvantage is the exposure of harmful alleles also, right? So harmful recessive alleles, to be more precise. So this is the correct answer. Let's move to the next question. The practice of mating between individuals of the same breed having no common ancestors on either side of the pedigree for 4 to 6 generations is called as what? It's called as outcrossing. Okay, and the other one was cross breeding. Let's move to the next question. In MOET, that is multiple ovulation embryo transfer technology, a superior female is administered with hormones having FSH-like activity for yielding 6 to 8 eggs, okay, per cycle instead of just 1 egg, which is normally done. This is followed by artificial insemination or mating with an elite bull. The resulting embryo at dash cell stage, that is 8 to 32 cell stage, is recovered non-surgically and transferred to a surrogate mother. The female is set for another round of superovulation. So we see here it's going to save our time instead of the natural breeding and then gestation and all that. It's a very long time consuming process. So we do this, thereby making the female ready immediately for the next round of superovulation, right? So that is called as multiple ovulation embryo transfer technique. Let's move to the next question. Select the correct option representing the set of edible marine fishes. So here again, Hilsa, sardines, mackerel and pomfret are the marine edible fishes. I think we had a similar question popping up in NEET 2016 phase 2 exam, right? So that was a very tricky question. Next, so these three are common carp, katla and rehu. These are freshwater. We need to remember this. These are freshwater, right? And the others are mix and match. So let's move to the next question. The second last question. Hisardale is a new breed of sheep developed in Punjab by carrying out the cross between Bikaneri ewes and merino rams. We must remember this. It might get confusing when they swap the words here and there, but we must remember that it is Bikaneri ewes and merino rams, right? Next question, and the final question for this zoology test is: an industry devoted to the catching, processing or selling of fish, shellfish or other aquatic animals is known as simple fishery. It's not aquaculture or pisciculture or sericulture. Okay, so the correct answer over here is fishery. So this is about it. I hope you have understood the entire series of questions that we have discussed for minor test 4, and I hope and I really wish and pray that all of you have done remarkably well in this test. So wishing you all the best for now and for future. We will meet again shortly. Thank you so much. Have a great day, everyone.
Hello students, how are you all? Today we are going to discuss about this AIRTS, which is minor test 4, and here we will be discussing about botany portion. Now students, these are some of the details of the AIRTS which has started on the 28th November. It was started on 28th November. Now we are dealing today with the minor test 4. See here students, this is the schedule here. Today we are going to talk about this test which was done on 9th January. Here we are going to discuss about botany part. The first question here is, soma clones are obtained by plant breeding, irradiation, genetic engineering and tissue culture. Remember students, when we talk about the soma clones, first we have to see here the term. What do we understand by these soma clones? Basically, the somatic cells and clones of that. So we are producing the clones, obviously in the tissue culture here. Tissue culture technique, we are having the identical plants, genetically identical plants, as we can see here, will be produced from a plant part or one of the portion of the plant. So in this technique of tissue culture, we are having the production of soma clones. We can say that these are the soma clones, genetically identical parts here or the plants here. We are also calling them as plantlets. So first option, we are having here the D option correct. Another question here is breeding of crops to improve the levels of minerals, vitamins and proteins in them is known as. So here we are breeding the crops, and what is the purpose here? We are increasing the levels of minerals, vitamins and proteins. We are having the options here as micropropagation, somatic hybridization, biofortification and biomagnification. So remember students, whenever we are increasing the levels of minerals, nutrients, vitamins, we are fortifying it. One of the compound or one of the crop, we are fortifying it, adding here the minerals, vitamins, nutrients. So the term here, it will be biofortification. Breeding of crops, and the purpose is to increase the level of nutrients, minerals, vitamins. We are fortifying here these crops with these minerals, vitamins, etc. So we are having this option here is correct, C1, biofortification. So we are having example also here, which is golden rice. And this has been fortified, or the levels of what increased here? Vitamin A. So this is the one of the example of this process we are calling as biofortification. Another question here is, which of the following process or stages of sewage treatment produces biogas? So we are having the sewage treatment, and in what stage or at what stage or in which process we are having this production of biogas? We know that biogas, the main component here is methane. And we are having here options: primary treatment, aerobic tank, C is anaerobic sludge digester, and D is sequential sedimentation. So whenever biogas production is happening, it is happening in this stage, anaerobic sludge digester here. So obviously we are adding here anaerobes. Some of the microorganisms, they will be acting on this and they will be producing here methane. Remember when we were talking about the kingdom Monera, what we have studied there? We have studied there the methanogens that will be producing the methane here. See here we are having this representation. This is one of the stage of this sewage treatment. What we are having here? Methane production, or we can say that it will be utilized as biogas. And what was done here? Methane and carbon dioxide was produced by anaerobes, or the microorganisms that were acting on it. So in this process, we are calling as anaerobic sludge digester of the sewage treatment. We are having the production of biogas or methane. So C option is correct here. Another question It is having two statements here, statement A and statement B. Statement A is saying in paddy fields, cyanobacteria serve as an important biofertilizer. So what we are talking here about the paddy fields or the rice fields here. Cyanobacteria, they are working as an important biofertilizers. Obviously, they will be adding minerals, nutrients to these crops. So we are having here cyanobacteria. This statement is correct because Anabina and nostoc. These are some of the examples that will be acting as biofertilizer. So this statement is correct. A statement here is correct. Statement B is main sources of biofertilizers are bacteria only. It is saying that only bacteria, they are working as the biofertilizers. This is not true. Why? Bacteria as well as cyanobacteria. In the first statement, we have also said that cyanobacteria as well as the fungi. So not only bacteria, they are working as the biofertilizers. Obviously here cyanobacteria, fungi, they will be also working as the biofertilizers. So statement B here is incorrect. So we are having the correct option as A. Only statement A here is correct. Another question here is which of the following statement is incorrect? We have to identify the incorrect statement out of these options here. A is saying many members of genus Glomus form lichens. So remember students, when we were talking about the lichens, they are the composite organism. Obviously, what they are composing of? They are composing of here fungi and algae. Now these are the components of the lichens, and it is saying that the fungi, it is belonging to the glomus. So this is incorrect here. Why? Because fungi of mainly ascomycetes, they are forming this association of lichens here. When we were talking about the glomus, it is included in mycorrhiza. So this statement is incorrect. If we see other statements here, Azotobacter is a free-living nitrogen-fixing bacteria. Obviously, this one is correct. Azotobacter, it is not in symbiotic association. It is free-living and working in the nitrogen fixation. Root nodules is formed by symbiotic association of rhizobium with roots of legumes here. So obviously, we have talked about it, rhizobium. They are forming this symbiotic association with the nodule and residing in the nodules of leguminous plants. D is saying plant having mycorrhizal association show resistant to root-borne pathogens. This is also correct. Mycorrhiza, it is associated with this glomus here, and it is also giving resistance to root-borne pathogens. So our option here A is correct. Another question here is, which of the following is modified by genetic engineering and is used as a clot buster? So, which is we are calling as clot buster enzyme. We are having the options A. Lactic acid, B. Streptokinase, C. Statins, and D. Cyclosporin A. So, clot buster enzyme which has been modified and extracted is streptokinase. From where it is obtained? It is obtained from Streptococcus. So, streptokinase working as clot buster enzyme and it has been obtained from Streptococcus. So, B option here is correct. Another question here is, which of the following wheat variety is developed having resistance against leaf and stripe rust? So this leaf and stripe rust is the disease which is caused by fungi here. which is caused by fungi here, and we are having some options here: Pusa Swarnim, Pusa Shubhra, Pusa Komal, and Himgiri. Himgiri is the wheat variety which has been developed against this disease of leaf and stripe rust here. So D option here is correct. Another question having the two statements, statement A and statement B. Statement A says that life cycle of ectoparasites is more complex due to its extreme specialization here. So we are here talking about the ectoparasites. So they will be living outside or inside the organisms. Obviously, we are talking here about the ectoparasites. They will be living here outside. And it is saying that they are having extreme specializations. This is not correct for the ectoparasites. It is correct for the endoparasites here. So statement A is incorrect here. Statement B is saying that majority of the parasites don't harm the host organism. Is it true? Obviously, students, this is incorrect. Majority we are here talking about. When we talk about this interaction, parasitism, here we are having parasitism interaction which will be having this relationship, plus minus. So obviously here the parasite will be benefited, whereas a host, it will be having its disadvantages due to this interaction. Parasitism is having this interaction plus minus. So obviously here statement B is also incorrect. Majority of the parasites, they will harm the host here. They are not having this relationship of unaffected host will not be there. So we are having correct option as D. Both statements A and B are incorrect. Another question here, presence of thick cuticle on leaf surface or the sunken stomata and CAM photosynthetic pathway are adaptations associated with. So we are having thick cuticle, we are having the sunken stomata and the CAM photosynthetic pathway. What do we mean by this CAM photosynthetic pathway? It is cressulation, acid metabolism. And it is present in the species, in the plant species which are present in the dry habitat, which are the xerophytes here. So we are having this, these adaptations, certain they are given here. Sunken stomata, thick cuticle on the leaf surface. So obviously the plant species which are living in this dry habitat, they are having these adaptations. We are having the options here: hydrophytes, halophytes, xerophytes and the mesophytes. So obviously C option is correct here. Xerophytes are the plant species which are living in the dry habitat. They will be showing these adaptations here. Another question here is, read the following statement and mark the correct option by stating which one of is true and which one is false here. So we have to see the statements here and we have to identify here which statement is true and which one is false. So first statement says that according to Allen's rule, mammals from colder climates generally have shorter ears and limbs to minimize heat loss. So Allen's rule is saying that it is talking about the mammals. And from the colder climates, they will be having shorter appendages here. It is talking about the shorter ears and limbs. So appendages in these mammals which are living in the colder environment, they will be short and it will minimize the heat loss. Obviously the surface area, it will be decreased, as in this case. You can see here some species, they are from the tropics and some are from the colder or polar areas here. So they are having large appendages which were present on the warmer climate. They are having the warmer climate, their appendages, legs, ears, they are quite large than the species that were present in the colder environment. So Allen's rule here is correct. B is saying that, or the second is saying that in the polar seas, aquatic mammals like seals. We have seen the polar sea aquatic mammals. For example, the seals, they have thick layer of fat, that is, we are calling it as blubber, and below their skin that acts as an insulator. So obviously it is minimizing the heat loss, acting as the insulator because they are present in the colder environments. They are increasing the loss of body heat. Obviously this statement is incorrect here because it is saying that it is increasing the loss of body heat, whereas the blubber is acting as an insulation. Obviously this will decrease the loss of body heat. Another statement here is, so second one is incorrect. Third statement is saying that to cope with altitude sickness, physiological adaptations allow some organism to respond quickly to a stressful situation. This statement is also correct. We are having the altitude sickness and there are certain physiological adaptations that counter this altitude sickness. For example, we are having the increase in red blood cell count. So this statement is also correct here. Fourth statement is saying that desert lizards have physiological ability to cope with extreme temperature but cannot maintain body temperature by behavioral means. But they are adapting to these conditions obviously by the behavioral means, as we have seen in the case of hibernation in the colder areas and estivation in these dry or hot areas. As the desert lizards, they will be living in the dry and hotter areas. So they are adapting to these climates because of their behavior. So this statement is also incorrect here. Second and fourth statement, they are false here. And first and third, they are true here. So we are having here, A is saying that, which one is saying first and third, they are true, and second and fourth, they are false. So obviously B option is correct here. Another question, which of the following process or processes contribute to increase in population density? So we are having the population density and increase. We are talking about which one of the attribute will contribute to increase in this population density. We are having the mortality only, mortality plus natality, natality plus immigration, and natality plus emigration. See here, we have to see the population density, it will be increasing. Now see here this representation. Population, we are having the population density we talk about. We are having the natality, immigration, mortality and emigration. Mortality we are having when we are having the death of organisms. So obviously the population density will decrease here. Emigration, that is when a population or the species, they are going to some other area. So these two attributes, they are decreasing the population density here. And only natality and immigration. Whenever the organisms, they are coming to this area, they are contributing to this area, it will be having increase in the population density. So we are having natality, that is the organisms, they are taking birth here, and immigration, when the organisms, they are moving from one area to another area, whatever the population density here we are talking about. So we are having correct option as C. Natality and immigration, they will increase the population density. Another question, urn-shaped age pyramid, it represents A, stable population, B, declining population, C, expanding population, and D, old population. See here students, we are having these three shapes of the pyramids. Expanding when the pyramid, it is this shaped, and stable when it is this bell shaped, and urn shaped when it is declining. So obviously the population here will be declining. But why this is happening? Because the pre-reproductive phase and the post-reproductive phase, they are quite smaller here. And that is why we are saying that pre-reproductive phase is very small. That is why the population, it is declining and it is having this shape. And whenever we talk about the bell-shaped, we are having the declining post-reproductive phase. So obviously it will be bell-shaped here, and a pyramid-shaped we are having, it is expanding because we are having here pre-reproductive population which is higher in number. So we can say that it will be declining population. Whenever we are representing a population in this urn-shaped pyramid, we will be having the declining population basically based on this pre-reproductive phase population which is very small in number. Another question, amount of biomass or organic matter produced per unit area over a time period by plants during photosynthesis is known as. Here we are talking about the amount of biomass or organic matter that will be produced per unit area over a time. And it is because of the process of photosynthesis by the plants. So what we are calling this term as nutrient cycling, primary productivity, secondary productivity, and community productivity. So obviously here, what kind of organisms they are taking part? They are producers here. And here we are talking about the production of these producers. So obviously we are calling it as primary productivity. Another question here, having the two statements, statement A and statement B. Mollusk is a common herbivore in the aquatic ecosystem. So mollusks, they are acting as a common herbivore. So obviously they will be feeding on the producers. Another statement says that energy trapped into an organism remains in it forever. This is incorrect, obviously. Energy is being transferred from one trophic level to the another trophic level. So we cannot say that energy, it will be trapped in an organism and it will remain there itself. So obviously this statement B is incorrect, and we are having the statement A is correct here. Option A, only statement A is correct. Another question, mark odd one out with respect to the trophic level of an organism. We are having the phytoplanktons, grass, trees, and grasshopper. So remember students, when we are talking about the trophic levels, we are having the producers, then we are having the consumers. So obviously the primary consumers, herbivores, they all come under this category of consumers. So we are having A, phytoplankton, B, grass, and C, tree. So obviously these three are producers here. Phytoplanktons, they are planktonic species of an aquatic ecosystem. And grass and tree, they are all producers. They are capturing the sunlight and will transfer the energy to another trophic level. Whereas the grasshopper, it is a herbivore here. It is one of the consumer. So this is odd one out, because others, they are at the producer trophic level, whereas the grasshopper, it is a herbivore which is a consumer. So we are having the correct option as D. Another question, choose the correct sequence of stages in a hydrarch. A saying submerged plant stage, marshmallow stage, and reed swarm stage. B, marshmallow stage, scrub stage, and forest. Scrub stage, phytoplankton, zooplankton, reed swarm stage, and phytoplankton and forest. Whenever we are talking about the ecological succession, what we are having here, ultimately succession will lead to a stable community here. So stable community, we can talk here about the forest and here. So other options, they are eliminated here. Obviously the forest, they are a stable community in comparison to the reed swarm stage or the zooplanktons here. So if we talk about the B and D option, marshmallow stage, scrub stage, reed swarm stage, and phytoplankton. Obviously these are the producers. So we cannot say that this one is correct. Obviously here the correct option will be B. Marshmallow stage, scrub stage, and forest here, the stable community. You can see here also, we are having this hydrarch. It is going in this direction. These are stage, marshmallow stage, and then it will come the scrub stage, and ultimately we are having a stable community, that is the forest here. If we talk about this representation also, it is also showing a hierarchy or the ecological succession in the aquatic ecosystem. We are having the pioneer stage, summer stage will come, floating stage, reed swarm or emergent stage, and after that sedge meadow stage, woodland stage, and ultimately the climax forest community, which is mesophytic in nature and which is quite stable community. Another question here, statement A is saying species composition and stratification are two main structural features of an ecosystem. So species composition here and stratification. We are having the stratification with respect to the habitats here. We are having in a forest different kinds of stratas here. So obviously these are the main components of ecosystem. Another statement is saying biotic community is dynamic and undergoes changes with the passage of time. Obviously the biotic community, it is quite dynamic, changing over time. Or we can say the evolvement of some species community, it is happening here. So statement B is also correct. We are having both the statements A and B, they are correct. Another question, the second trophic level in a lake usually, A is phytoplankton, zooplankton here, benthos and fishes here. We are talking about the second trophic level in a lake, in an aquatic ecosystem here. So phytoplanktons, they are producers, planktonic species obviously. Zooplanktons, they are also the planktonic species, but they are on the consumer level here. Second trophic level after the phytoplanktons, it will be occupied here with the help of these zooplanktons. So B option here is correct. Another question, when a host fish species become extinct, its unique assemblage of parasites also meets the same fate. So we are having the extinction process and host fish species, it is becoming extinct, and what is happening after that? Parasite is also becoming extinct. The parasite, it was wholly dependent on this species of fish. So both are going extinct, and it will be called as, we are having the options as alien species invasion, overexploitation, habitat loss and fragmentation, and co-extinction. So obviously this will be known as co-extinction. Why? Obviously this was the interaction we were having, parasitism, as the parasite was involved here. Whenever the parasite which was completely dependent on this fish species, it is getting extinct, and because of that the parasite, it was getting extinct. So we can say that co-extinction is happening. Another question, select the mismatched pair. Robert May global species diversity, Alexander von Humboldt species area relationship here, Paul Ehrlich competitive exclusion principle, Robert Costanza ecosystem services. So here Robert May has given this global species diversity, and von Humboldt, he has given species area relationship. But when we talk about the competitive exclusion principle, it was given by Gauss. We are having this mismatched pair here, whereas Robert Costanza, he has given the ecosystem services. What is the value of our ecosystem here? And we have explained in the ecosystem services. So C option is the mismatched pair here, and C option is correct. Another question, which of the following statement is correct? We have to identify the correct statement here. A is saying presently 23% of all amphibian species in the world, they face threat of extinction. They are going to extinct after some time. So we are talking about the amphibian species, but the percentage here is not correct. They are 32%. 32% of all the amphibian species, they are having this threat of extinction. Speciation is generally a function of surface area. Is it correct? Speciation, that is the evolution of species. Is it the function of surface area? Obviously it is not the function of surface area, it is the function of time. Another option, extinction of passenger pigeon was due to overexploitation by humans here. Obviously, this statement is correct. India has more than 50,000 varieties of mango. When we are talking about the species diversity, we have seen that mango, it is having various varieties here. So obviously this number is not correct. It is having 1500 varieties of mango. So C option here is correct. Another question, exploring molecular, genetic and species level diversity for products of economic importance. So we are having here application of molecular, genetic and species level diversity and how they can be utilized for our benefit, for economic importance. And we are talking them about in bioremediation, obviously not, biofuel, biomolecule, bioprospecting. Bioprospecting is the field where we will exploit here molecular, genetic and species diversity, and they will be utilized for the humans. So we are having the correct option D here, bioprospecting. In that, we are having the application of this diversity. Another question, when we conserve and protect the whole ecosystem, its biodiversity at all levels is protected. Choose odd one out with respect to the example of this approach. Whenever students we are talking about the biodiversity conservation, we are having two methods and what they are known as ex situ and in situ. And when we talk about the ex situ kind of biodiversity conservation, we are having in that the organism, it will be protected outside its habitat. So obviously the ex situ, we are not having this term. It is saying that when we are conserving and protecting the whole ecosystem, here we are protecting the whole ecosystem. Where it will be counted in the in situ kind of method of biodiversity conservation. So it's at all levels the biodiversity, it will be protected. And we have to see which comes under this in situ and which is not coming under this in situ kind of method. We are having the options biosphere reserve, it is coming under this in situ kind of conservation, wildlife safari parks. Obviously in this we are not conserving the organism in its habitat. The whole ecosystem is not protected here. So we are having the correct option here as wildlife safari parks, which comes under ex situ. Wildlife sanctuaries and national park, they come under this in situ method of conservation. So we are having the correct option as B. Another question, presence of large amounts of nutrients in water causes excessive growth of free floating algae. And this is referred to as, we are having the first term eutrophication, B is biomagnification here, algal bloom and bioremediation here. So whenever we are having, for example, surface runoff we are having, in that we are adding a lot of phosphates in the ecosystem, aquatic ecosystem, amounts of nutrients, they will be added in the water. And what it will cause excessive growth of free floating algae. And this is known as algal bloom. Because here the algal, we are having the enormous growth of algae. As you can see here, the water, it has turned green because of this algal bloom. Magnesium, phosphate salts, they are adding in this water and they are causing a nutrient-rich environment, and because of that algae, they will be having very enormous growth and turning the water green here, and it is known as algal bloom. Another question, polyblend is a refined powder of bitumen, recycled modified plastic, charcoal, and D option says charcoal plus plastic. So if we talk about the polyblend, it is recycled modified plastic and it has been used in the formation of the roads, making them better and ready for adverse situations, or we can say that they are better suited for different climates, we can say. Rainy season, they are quite resistant to certain conditions. So we are having the polyblend, which is a result of recycled modified plastic. Another question, which of the following is the only solution for the treatment of e-waste? Whenever we talk about the e-waste, Obviously, the waste we are generating from the electrical appliances here, electronics we can say. So what is the ultimate remedy for this e-waste? We are having certain terms here, reducing. We cannot reduce the application or usage of these electronic items. Obviously, reducing is not the method by which we can reduce the e-waste. Reusing, they cannot be, they can be to certain extent reused, but ultimately recycling is the method by which we can reduce the amount of this e-waste generated here. So C option is correct. Another question, which of the following statement best describes parasitism? Remember, I have also told you parasitism, a kind of interaction between the species when we are having this relationship. Here the parasite, it will be benefited, whereas the host, it will be having adverse effect on itself. So this kind of interaction is parasitism. It is saying that both the organisms are harmed. We are not having this relationship in this parasitism. Both the organisms are benefited. No, this is not mutualism here. C saying one organism is benefited while the other is not affected. This is not the situation here. Whereas D is talking one organism is benefited, yes, and while the other it will be harmed here. So we are having this relationship in parasitism. So D option here is correct. Another question, which of the following is not the effect of ozone depletion? So ozone depletion, it is harming our environment as well as the humans. So we are having certain characteristics or certain effects of this ozone depletion. What is having here? Snow blindness, obviously it is the effect of ozone depletion. Skin cancer, cataract. Obviously these are the three effects of this ozone depletion. As the thinning of ozone is happening, and ultimately we will have the penetration of the harmful rays of the sun. So because of that, we are having certain diseases here. Whereas the D option is saying that El Nino effect. El Nino effect is when we are having abnormal rise in temperature of the sea or the environment above it. So we are having this El Nino effect, and this is because of the, we can say that, climate change. Climate change is causing this El Nino, La Nina effect, and we are having this D option, which is not the effect of this thinning of the ozone or the ozone depletion. Another question, Ozone Day is celebrated on 16th of August, 16th September, 16th October and 16th November here. So B option here is correct. Ozone Day is celebrated on the 16th of September. Another question, lice on humans, ticks on dogs, marine fishes infested with copepods, cuscuta growing on hedge plant. Which of the following population interaction is shown by above example? So lice on human, whether the lice will be affected, obviously it will be benefited. Humans, they are harmed or they are not affecting. See here, ticks on dogs, marine fishes infested with copepods. So some organism here is being affected, whereas the some organism, another organism, it is being harmed here. Another is saying cuscuta growing on hedge plant. So obviously these are all the cases of parasitism, which will show here the plus minus relationship. One organism, it will be benefited, whereas the another organism here, it will be harmed. Another question, match the following. Column A is having certain options here, column B is having certain. Nitrogen cycle, we have to match from it. Dobson unit, sedimentary cycle, gaseous cycle and decibel. Obviously nitrogen cycle, it goes for gaseous cycle. Phosphorus cycle, it is also one of the biogeochemical cycle, which is sedimentary in nature. Remember, biogeochemical cycles, they are of two types. sedimentary and gaseous. So gaseous is example for, we are having nitrogen cycle, and sedimentary phosphorus. Basically, it's a repository as phosphates, sediments. Noise, we are having the remaining options Dobson unit and decibel. Noise is calculated in decibels. Ozone thickness, it goes for Dobson unit. So nitrogen cycle third and phosphorus second. First, third and second is for second. These two options are having, whereas the noise 3-4 decibel. So obviously the C option here will be correct. Ozone thickness, it is measured in Dobson unit. Another question, which of the following international treaty was signed to control the emission of ozone depleting substances? ODS. Students, remember when we are talking about the ODS, we are having one of the examples as chlorofluorocarbons, CFCs. So a treaty was signed to control the emissions of these ozone depleting substances, which is affecting the ozone and thinning it and causing it ozone depletion. So we are having the World Summit on Sustainable Development, the Earth Summit, Montreal Protocol and Basel Convention. Obviously C option here is correct, which was signed, a treaty was signed to control these ODS, ozone depleting substances here. Another question, in which of the following population interaction fitness of one species is significantly lowered in the presence of another species? So we are having one of the interaction, these kind of interactions are given. We have to see in which interaction we are having this interaction here. Mutualism, obviously we are having the plus interaction. Predation, parasitism and competition are remaining. So obviously in the competition, because the two species here, they are fighting for similar resources. They are getting affected. They are getting affected negatively here, both the species here. So obviously competition is one kind of interaction in which we will see minus kind of interaction. So D option here is correct. Another question, it is having two statements. Statement A is saying electrostatic precipitator can remove over 99% particulate matter present in exhaust of thermal power plant here. So what it is doing? Over 99% of the particulate matter it is removing. This is correct for electrostatic precipitator. As you can see here, this is the representation of electrostatic precipitator. Utilizing here the corona negatively charged ions here and the positively charged ions here. Statement B is saying that motor vehicle equipped with catalytic converter should use unleaded petrol here. Obviously this statement is also correct, but why we are using here unleaded petrol? Because lead, it interferes with the activity of these catalyst. What kind of catalyst we are using in this catalytic converter here? Rhodium we have used, palladium we are using, and whenever we are using this leaded petrol, it will be affecting this efficiency of this catalytic converter here. They will be affecting these catalysts we are using as rhodium and palladium. So obviously we have to use here unleaded petrol. So only statement A is correct, only B, both statements are correct, and both statements they are incorrect. Obviously here C option is correct. Another question, which of the following statement is incorrect here? We have to identify the incorrect statement. DFC begins with dead organic matter. See here, this is the example of DFC here, a kind of food chain we are having. Now some of the organisms of GFC are prey to DFC organisms. This is incorrect here. This is the vice versa we are having. Another statement saying that decomposers are heterotrophic organisms, mainly fungi and bacteria. This is also correct. Fungi and bacteria, they are acting as decomposers in this DFC, and they are the heterotrophic organisms. And it is starting with the dead organic matter. As you can see here, the dead leaves, they are eaten by some insects here, and again we will have certain microorganisms. GFC is the major conduit for energy flow in an aquatic ecosystem. So grazing food chain we are also having there. So here B option is correct because this is the incorrect statement. Another question, the evolution of two interacting species confer benefit on both and they are tightly linked with one-to-one relationship. So we are having here evolution of two interacting species. They are conferring benefit to each another and they are tightly linked with one-to-one relationship. Which of the following example best explains the above statement? Abingdon tortoise and goat, Balanus and Thymallus here. So obviously these are the examples of competitive exclusion as was given by Gauss. Whereas if we talk about the C, fig and wasp, it is showing this plus kind of interaction. As we have seen in the statement, we can say that symbiotic association or mutualism is being shown by them. So C option here is correct. Whereas D says that marine fish and copepods. So obviously we are having this kind of interaction in it. So marine fish and copepods, this is the example of parasitism here. So C is the example of mutualism which is explained by this statement, fig and wasp. Another question, choose odd one out with respect to the population attributes. So we have to see the population attributes here. Sex ratio, age pyramid, death rate, and we are having the birth here. When we talk about the population attributes, as we have seen in the case of natality, immigration, emigration, and the mortality, we are talking about the rates. So this is not correct here, D. Birth rate, we are talking about it. So obviously the D option here is correct. This is not the population attribute. If it would have been written like birth rate, it was having the population attribute. Another question, all are alien species whose invasion may cause disappearance of indigenous species through changed biotic interaction except. So we are talking here about the alien species invasion. And it has caused the loss or the extinction of another species where they have invaded. Because they have come from one community or the one habitat, they have entered a new habitat where they have caused the extinction of that, we can say that related species which was present earlier there. So we are having examples, water hyacinth, Lantana camera, Parthenium, and the solanum. So all these three, A, B, C, they are the example of alien species invasion, which has caused the biodiversity loss or the extinction of some species. Whereas if we talk about the D, solanum, obviously this is the scientific name for potato. This is not the example of alien species invasion. Whereas in India, it has came from the South America, but this has not caused the extinction of another species which was present in India. So potato is odd one out here. D, we are having the correct option. Another question, how much solar energy is received at third trophic level if this amount of joules of sunlight is available to primary producer? So this was incident, we can say that, on the producer. See here we are having this representation. Now see here students, sunlight, it was given this. But I have to tell you here that we are talking here about the sunlight. We are not talking here about the power. Because it is sunlight, 1 to 5% of the sunlight and 2 to 10% of power. It is taken up by the producers. So after that, what we will be getting, as it is sunlight, only 1 to 2% of it will be taken up by the producers, primary producers. So we are having this. Why? We have taken the 1% of this amount of joules, as it was sunlight, not the par here. If it would have been par, we have taken the 10% of it. So we can say that now we are having the available amount as this amount of joule. And we have to see that third trophic level it will be having. If I calculate here 1%, or I can say that of this amount of joules. So we are having here now at the first trophic level. What I will be having here this amount of joule. Now we know that according to Lindman's rule, we are having the 10% of the energy, it will be transferred to the next trophic level here. 10% rule. And why it is not applying here, why it is not applied on the sunlight? Because 1 to 2% of the sunlight will be taken up by the producers, or to 2 to 10% of the power it will be taken up by the producers. We have given here the sunlight, so only 1% we have taken here. After that, 10% of this available at the first trophic level, it will be taken. Now at the second trophic level, I am having here this amount of joules. Now at the third trophic level, I am writing it here, third trophic level, 10% of this, what was present on the second trophic level, now we are taking here. So we are having on the third trophic level 100 joules available. So we are having the correct option as C. Only here the trick is 1% we have to take from the sunlight. If it would have been given for the power, we would have taken 10% here. So after that 10% we have taken. Now we are getting at the third trophic level 100 joules. After that 10% of it will be at the fourth trophic level. So our answer here is 100 joules. Another question. Choose correct options with respect to the pollutants stipulated in fuel according to Euro 3 norms. We have to see Euro 3 norms. It has regulated the pollution here, certain pollution policies here. So we are having the options. Diesel 350 ppm of sulfur. So it was destined to reduce the sulfur amount in this diesel and petrol. This is correct for Euro 3 norms. If we talk about the petrol, it is at 50 ppm of sulfur. It aimed at reducing the sulfur content in the petrol to this 50 ppm. This is not correct for Euro 3 norms. It was at 150 ppm. Aromatic compounds, it was at 92%. This is not correct for Euro 3 norms. It aimed at 42%. D, both A and B. So obviously this is incorrect here. We are having the correct option as A. Another question, the product of Monoscus purpureus has been commercialized as. So we are having this yeast or microorganism. Now we have exploited certain microorganisms and we have obtained certain effective chemicals or compounds from that. Now the product of this Monoscus, is it a clotbuster, bottle juice clarifying agent, blood cholesterol lowering agent, and immunosuppressive agent here? So obviously the Monoscus has been utilized in the production of this blood cholesterol reducing agent, which we are calling as statin. So C option here is correct. Another question, which of the following is correct for R selected species? So R selected species and K selected species, they can be represented with here with this graph also. So K selected is dependent on the carrying capacity. Whereas the R selected species, they are having what characteristics we have to identify here. Produce large number of progeny with small body size. Produce large number of progeny with large body size. Produce small number of progeny, small body size, and produce small number of progeny with large body size. So these two statements, they are incorrect because in R selected species, we are having little survival. after certain age, they will not have, they are having very lower survival conditions. So obviously the A statement here is correct. They are producing the large number of progeny which are having very small size, as you can see here. And they are having very little chances of survival. So we are having the correct option as A. Another question, statement A and statement B. Statement A is saying that coral reefs are primary producers of the deep sea hydrothermal vent ecosystem. Deep sea hydrothermal vent ecosystem. It is having the producers which are chemotrophic bacteria, not coral reefs here. And when we talk about the chemotrophs, so obviously they are utilizing chemical compounds here to synthesize their food. So these are the chemotrophic bacteria which are acting as producers in these extreme conditions, deep sea hydrothermal vents. They are having extreme high temperatures. So statement B is saying that lichens appear as the pioneer organism during succession on bare rocks. So obviously this statement is correct. Lichens, as we have seen, which is the association of fungi and algae. They are acting as a pioneer species. They will establish themselves on the bare rocks and will help in the ecological succession. In the primary succession, we have seen that. So the only statement A, B is correct. Both are correct and both. So obviously, only B statement here is correct, as coral reefs are not the producers of these deep sea thermal vents. They are the chemotrophs. Another question, population of birds declines significantly in an area where DDT is used extensively due to A, snakes were feeding exclusively on birds. When we are talking about the DDT, we are talking here about the biomagnification. One of the phenomenon we are having. So see here, students, this is the representation which is showing that we are having the increase in concentration of these chemical compound. We have seen the example of DDT. It is magnifying, or the concentration of this DDT, it is increasing with the increase in the trophic level. Increase concentration with increase in trophic level. So we are calling this phenomenon as biomagnification, and it is getting deposited here or having the higher concentration in the buds, affecting the eggshells. It is affecting the calcium metabolism in the buds and causing the thinning of the eggshells. So we are having premature breaking of eggshells because of this phenomenon of biomagnification, which is caused by, in this example, DDT. So we are having the correct option here as C. Another question, Jaya and Ratna developed for Green Revolution in India are the varieties of. So Jaya and Ratna we have obtained because of this Green Revolution, and they are the varieties of rice. C option here is correct. Another question, a biologist studied the population of rabbits in a field. He found that average natality was 250. So we are having natality as 250. Average mortality was 240. I can write it here. Immigration 20. So immigration and natality will add to the population. So obviously we are combining it as, and we are getting here 270. And mortality and emigration, they will decrease the population here. So mortality, as we have seen, it is 240, and the emigration, it is 30. It is adding up to 270. So will there be rise or decrease in the population? Obviously not, because the increase and the decrease, they are equal here. So we will not be having increase or decrease in the population here. So we can say that the net increase in population is 0. D option is correct here. You can see here, we have seen this representation also. Natality, immigration adding to the population, whereas mortality and emigration, they are subtracting the population here. Another question, statement A says that parasite is an organism which always live inside body of another organism and may kill it. Always lives inside. Is it correct for the parasites? Obviously not, because parasites, they can be endoparasites, they can be ectoparasites. So this statement is ultimately incorrect. Statement B is saying that a lion eating a deer and a sparrow feeding on a grain are ecologically similar in being consumers. So obviously, lion, it is eating a deer, and sparrow, it is feeding on a grain. This is the product of, we are having the producers. So obviously, these statements, they are correct. So we are having only B statement, which is correct. Another question, choose odd one out with respect to the process of ecological succession. We are having the process of ecological succession and it is having certain characteristics. As you can see here, we are having this process of ecological succession. This is our initial phase. Now the climax community we are having here. And these are the seral stages. Now these are the characteristics. Which one is odd one out? Species diversity, it increases. As you can see here in this representation also, species diversity, obviously it is increasing during the course of ecological succession. Structural complexity, it will also increase. As you can see easily here, structural complexity, it is increasing. Niche becomes unspecialized. Here, this statement is incorrect because we are heading to a stable community here. Climax community we are having, it is quite stable than other communities we were having in the ecological succession. So niche, it is becoming specialized here. Food chain relationship becomes complex. So obviously whenever the species diversity it is increasing, we are having the structural complexity also. Obviously the food chain relationship, it will become complex, it will be having increasing complexity. So C option here is correct. Correct here, as we are having this odd one out. Another question, choose odd one out with respect to the primary carnivores, secondary consumers in terrestrial ecosystem. Toad, spider, lizard, insectivorous birds and spider, toad and fishes, centipedes and lizards. So see here students, we are having this representation of our food chain in the form of pyramids. You can see here the autotrophs or the producers here, herbivores, primary carnivores and the secondary consumers. So it is talking about this level, primary carnivores here or secondary consumers here. When we talk about the fishes, they sometimes also act as secondary carnivores. They are feeding on the small fishes here, so they will also come at this level. So obviously this one is odd one out here. We are having the C option as correct. The last question here is, which of the following statement is correct with respect to the humus? When we talk about the humus, it is here we are having certain options. Light colored, obviously not, it is dark colored. And we are getting this humus because of the process of humification, as we have seen in the case of decomposition there. So B is saying that poorly resistant to microbial actions. It is highly resistant. C is saying that colloidal in nature. If we talk about this humus, it is obviously showing this nature. It is having this colloidal nature. So this is correct for humus. Whereas if we talk about the D, less decomposed or inorganic matter here. So obviously it is the organic matter because it has been produced because of the process of humification. So D option is also incorrect here. We are having the correct option as C. So students, this was all about the botany portion that we have discussed today. That was in the minor test 4. That's all for today. Thank you.
Hello everyone, welcome to the session. So over here, let's start with the discussion of chemistry portion of the AIRTS. So over here, first of all, let's see over here when our test series was started. Our test series got started on 28th of November, and this test series is specifically for 12th and 12th passed out students who are appearing for NEET 2023 exam. And this test series covers all the 4 subjects: physics, chemistry, botany, as well as zoology. And over here we actually had 4 minor tests, 2 mega tests, and 6 full syllabus tests, out of which some of the tests has been done already. This is totally based on the NEET pattern, having section A as well as section B, in which out of the 15 questions you have to attempt 10 questions. 5 questions are basically an optional question for you. So the medium is again English medium, and the time given to you is 3 hours 20 minutes as always. Now over here, minor test 1, 2, 3 was already conducted. Now recently minor test 4 had been conducted on 9th of January. Next minor test would be conducted on 23rd of January. So be prepared for that particular this test. This test is going to be a mega test, a big full syllabus test over here, conducted on 23rd of January. So today we are going to have a discussion based on the test which happened to be on 9th of January, this Monday. Alright, so let's start with the discussion, talking about your question number 51. So over here, what is given to us now, we are having phenol over here, phenol in presence of CCl4. CCl4 along with alcoholic KOH, when it is heated, this is basically a type of Riemann reaction where instead of salicylic acid is formed. When we use CCl4, what do we get? We get salicylic acid. But if chloroform is given to me in the reagents, then over here salicylic would have been formed, but over here salicylic acid would form. And then when I am going to treat this molecule with acid chloride, the acylation of the OH group will occur, and what am I going to get over here? The B molecule is going to be this. Now COH is already here. Now over here, instead of OH is going to get acylated, and I am going to have over here COCH3 group. So this is over here your aspirin. So my answer to this question is going to be option number A, which is salicylic acid, A product, and B is going to be your aspirin, which is this molecule. Alright, moving on to the next question, number 52. Now over here, this is an alkene, as the formula is suggesting to us. The formula is going to have a general formula CnH2n. So this is C4H8. Now when it is getting brominated, bromination is basically an anti-addition. Okay, so anti-addition will occur, and I am going to get 2 products, and these 2 products are enantiomers of each other. That is, I am going to get a racemic mixture. Okay, now you must have studied about, if I am going to get a racemic mixture over here. Now you must have heard about a simple formula, Carr and Tam rule, right? So Carr and Tam rule is when trans alkene undergoes an anti-addition, we get the meso product. So over here, we do not have a meso product, rather we are having a racemic mixture. So over here, Carr rule will be applicable whenever a cis alkene will undergo an anti-addition. It is going to give us racemic mixture, Carr rule. Cis on getting anti-addition product is going to give us racemic mixture. So answer is going to be, this alkene is basically going to be a cis alkene, right, having 4 carbon atoms. So I am going to have cis-butene. So answer is going to be only one option, that is option number A. Alright, moving on to the next question, number 53. Incorrect match is which of the following? Sucrose on getting hydrolyzed is going to form glucose and fructose. This is absolutely correct. So this is correct. Sucrose is made up of glucose and fructose. Lactose on getting hydrolyzed is going to form galactose and glucose. Again, it's correct. Maltose is made up of 2 glucose units. So mannose is not formed here. This is wrong. So this complete hydrolysis reaction is wrong. A and B is correct. Now, some of you have marked option D here, both A and B, but these are the correct options. In the question, they have asked you to mark the incorrect match. Okay, so answer is option C, incorrect. Maltose is made up of 2 glucose units. Moving on to the next question, number 54. Cetyl trimethyl ammonium bromide is a common what? What kind of detergent, it is non-ionic, cationic, anionic or antioxidant. Now over here cetyl trimethyl ammonium, as the name over here suggests, ammonium bromide. Now bromide over here is going to be a counter ion, and if the negative ion is your counter ion, that means your detergent is having a positive charge. Now over here if I'm having ammonium, that is it is cationic detergent. So answer is going to be option B, cationic detergent. Alright, moving on to the next question, number 55. Which of the following will not give Cannizzaro reaction? Now this is a very important exceptional part over here. Cannizzaro reaction is not shown from which of the following. Now for Cannizzaro reaction, there's one hypothesis that that in whatever compounds alpha hydrogen is absent, that they are going to show Cannizzaro reaction. But over here in this case, alpha hydrogen is present, then also it is going to show Cannizzaro. So it shows Cannizzaro, please mark this. It shows Cannizzaro, and why is that so? Because over here, what happens in Cannizzaro? If Cannizzaro is not happening, aldol would be occurring. If aldol is occurring here, that means this hydrogen will be removed and this is going to form over here an anion. And I am actually making the bonds of this carbanion to two methyl groups which are having plus I group, and hence they are going to destabilize this carbanion present over here. So it cannot undergo aldol condensation. So in presence of a base, it is going to undergo, or it is going to show Cannizzaro reaction, that is self-oxidation and self-reduction reaction. Okay. Now talking about CCl3CHO, definitely this is not going to show because alpha hydrogen is not present over here. Hydrogen is not present, chlorine is present. So this is not going to show Cannizzaro reaction. Over here, this is going to show Cannizzaro, this shows Cannizzaro, this also shows Cannizzaro because they are not having any sort of alpha hydrogen. Okay, most of you must have marked option A instead of B. So be careful about this. Over here, in aldol condensation is not possible because the carbanion is unstable here. Okay, next question, number 56. In the following reaction, X and Y respectively are which of these? Now over here I'm having an alkene in the presence of HBr. Markovnikov addition will occur directly. Markovnikov addition is going to make a more stable carbocation. So over here the allylic carbocation is more stable. So in the first case, in the first X product, over here I am going to get at this position, I will be getting a carbocation, and Br negative will attack at the allylic position only. But when I'm having in presence of peroxide, so in presence of peroxide, by default, the most stable free radical is not observed because free radicals are quite reactive. So they are going to attack on the least sterically hindered position, which is this position over here. So my answer to this question over here is going to be this. Bromine in the X case, bromine is going to attack on the allylic carbon atom, and over here this is going to attack on the primary carbon atom. So answer is going to be option B. Alright, next question, number 57. The major product of the following reaction is which one of these? Now whenever an acid chloride is treated with an amine, what happens? HCl is removed out. When HCl is removed out, there is a bond formed between NH and carbonyl, thereby making an amide linkage in between. Right? And I am going to get an amide out of it, and then further that product is undergoing bromination in the presence of FeBr3, which is a Lewis acid. Now this is going to undergo some sort of reaction which is responsible for the formation of Br positive electrophile. Okay, now if Br positive electrophile is generated, now this electrophile is going to attack on a particular position. Now first of all, what do we have over here? What is formed? Now I am having over here X is basically, X is this CONH, and I am having a benzene ring over here where at the para position methyl is present. Right? Now in this case I have to see that which ring is more activating ring, because only on that ring electrophile is going to move and attack. Okay, now electrophile is electron loving, so I have to check which ring is more electron rich. Now if I'll just talk about the ring which is bonded to carbonyl, now this is going to have a minus R effect, whereas this is going to have a plus R effect, thereby making this ring electron rich. If this ring is electron rich, that means bromine bromine cation is going to come and attack on this benzene ring, right? Either at the ortho position or at the para position. But in this case, para position is already substituted with the methyl group, so it is going to attack by default at the ortho position, right? So it is going to attack over here. So bromine plus ion is going to come and attack on the ortho position here, and the answer will by default be option A in this case. Okay. Next question, number 58. Which of the what is the major product of the reaction? So over here H2SO4 is there, which is a dehydrating agent. Over here water will be removed out. So it is going to remove OH negative from here, and it is going to attain a positive charge. So if this is going to get the positive charge, right. Now this is going to get a positive charge over here, right. Now this carbocation is actually a secondary carbocation, and I want to make this carbocation more stable. How I can do that? If I'll just do the 1,2 methyl shift. Now what am I getting over here? I am getting over here a tertiary carbocation. I have got over here a tertiary carbocation, and from this tertiary carbocation, now from here hydrogen will be eliminated, and thereby this double bond, I will be getting a double bond between these two carbon atoms, making it more alkylated alkene and hence a stable alkene. So answer is going to be by default option C in this case. Alright, next question, number 59. CH3, CH2, CH2, NH2 in presence of HNO2, nitrous acid, it is going to give us A as the major product. And then in presence of PI3, AgNO2 and again nitrous acid, it is going to give me B. Now this over here is your Victor-Mears test. Okay, let's read out the statements. For the given reaction sequence, identify the correct statement. Now please again highlight the word correct. You have to identify the correct statement. First option is A is CH3, CH2, CH2OH. Absolutely correct. This reaction is going to form an alcohol. So this is the correct statement. B is nitrolic acid. Absolutely, on reaction with PI3, AgNO2 and HNO2, this is going to form a nitrolic acid, which is again after getting solubilized in a basic medium or alkaline medium is going to give me a red color. So this statement is also correct. Now if you remember in Victor-Mears test, there is a rule there. For primary amines you are going to get a red color. For secondary amines you get blue color, and tertiary amines are colorless. At the end you get the colorless product. So RBC, using this I usually teach in the class, RBC for primary, secondary and tertiary respectively, red, blue and colorless. So for primary it is red colored, that means all these three statements are right. Answer is option D. Okay, question number 16. Which one of the following is Gilman's reagent? Now what is Gilman's reagent? Gilman's reagent is R2CuLi. That is dialkyl lithium copper. So lithium dialkyl copper, answer is option B. Alright, question number 61. Now this was a direct question. Identify the incorrect reaction. Now in this case you have to tell the incorrect reaction. Now first reaction over here is actually we are having a disonium salt. Disonium salt in the presence of H3PO2. H3PO2 basically removes everything which is present on the benzene ring. Okay, now we are getting phenol. Now this is wrong. On reaction with H3PO2, we should have get benzene, but this is wrong. So this reaction is wrong. In the presence of HBF4, fluorination is done, BF3 is liberated out, nitrogen gas is liberated out. This is absolutely correct. Third, now over here HBF4, NaNO2, no, this is not correct because this is right over here, NaNO2 and copper, this is making it wrong. Fluorobenzene will not be formed with this. So incorrect reactions have been asked to us. So answer is option D, both A and C reactions are incorrect. Alright, question number 62. The diketone over here on intramolecular aldol condensation gives the final product as what? So if I'm having this over here, now intramolecular aldol condensation means that in the same molecule aldol condensation would be occurring. One carbon atom being an alpha carbon will be removing alpha proton from it, and the anion is going to attack on the carbonyl carbon atom. right? So over here, this is going to, let's say from here, hydrogen has been gone and I have got negative charge here, and this is going to attack at this position. Okay, and now I'm going to get 1, 2, 3, 4, 5. 5-membered ring. 5-membered ring where I'm having a carbonyl compound, and over here, this is going to come up here having OH, and at the end, when I'm going to heat this up, OH and H from water will be removed out because alpha beta unsaturated product is formed in the aldol condensation reaction, and 1-methyl is bonded to this. So answer is going to be a 5-membered ring with a carbonyl compound or with a carbonyl group over itself with a double bond and a methyl group. So that is present in just one case, that is option C. Okay. Moving on, next question number 63. Which of the following reagents can convert nitrobenzene into phenyl hydroxylamine? So what are we doing over here? Nitrobenzene is getting converted to phenyl hydroxylamine, NH-OH. Now this is a characteristic reaction, specific reaction over here, which is done in the presence of zinc in ammonium chloride. So please, these are the reagents over here. Mark this is important. So this is the answer, option C. Alright, question number 64. Ethyl acetoacetate reacts with 1 mole of methyl magnesium bromide. Then product of the reaction will be what? Now I am over here talking about ethyl acetoacetate. Now what is ethyl acetoacetate? This is ethyl acetoacetate. Okay. Now, in this case, if I am treating this with 1 mole of methyl magnesium bromide, methyl CH3 magnesium bromide. Magnesium is going to have a positive charge, methyl would be getting the negative charge. Now, negative charge goes where? Negative charge basically goes on the electrophilic center. But in this case, if I'll just talk about this, now over here, if I'll just talk about this carbon atom. This carbon atom is surrounded by this carbonyl, as well as over here I'm having an ester group, right? COO group. So this is making this proton or this alpha carbon, protons attached, hydrogens attached to this carbon atom are basically very much more acidic. So from this carbon atom, the liberation of H plus ion is quite easy. So this is going to easily form CH negative here. So what am I going to get over here? CH negative. Now to this CH negative, what is going to come? MgBr positive will come. Instead of CH3 negative, this time MgBr positive is going to attack on the position from where the acidic hydrogen is being liberated. So answer to this question is going to be option C, because magnesium bromide is attacking here. Alright. Next question. Now, where is methyl going? CH3 negative going. The hydrogen, the H plus, which has been liberated out from here, we are going to get, that will be attacking over here, and I am going to get methane as a byproduct here. Alright. Next question, number 65. The end product in the following sequence of reaction is what? Now, I'm having over here cyclohexene in presence of ozonolysis. Now, please mark this important, because in ozonolysis, we are having water. With the water, this is going to be oxidative ozonolysis. There are two different types of ozonolysis. One is reductive ozonolysis, one is oxidative ozonolysis. When reductive ozonolysis occurs in the presence of zinc and HCl, in reductive ozonolysis, aldehyde is formed as a product. But in the oxidative ozonolysis, oxidized product is formed, that is carboxylic acid is formed. Alright, so I am going to get over here carboxylic acids. Over here also carboxylic acids. Now in presence of calcium hydroxide, a salt will be formed and ultimately calcium carbonate. From at this position, calcium carbonate will be removed out, one carbon atom will be less now. Now at the end, after this, at this point, I am going to get 5-membered ring. I am going to get a 5-membered ring here with a carbonyl group, and over here this is again a reducing agent which reduces the carbonyl into hydrocarbons, and then I am going to get ultimately cyclopentane directly. So answer is going to be option B in this case. Alright, next question, number 66. The number of amines possible for the molecular formula C3H9N are. Now over here C3H9N, that is I'm having 3 carbons, 1 nitrogen. Now what are the different isomers which are possible over here? Let's draw. So first is going to be normal primary amine, propanamine. Second, again, another type of primary amine that I can form over here is the position isomer of this, where I can just set amine at this position, at the second position. Right, so this is going to be propane 2-amine. Then the third one is going to be secondary amine here. Right, so this is your secondary amine. This is also third isomer. Now fourth isomer, if I want to draw over here, this is going to be tertiary amine. So this is primary amine. This is also primary amine. This is secondary amine. This over here is tertiary amine. So in total, I can make 4 isomers out of it. So answer is going to be option D here. Alright, next question, number 67. Which of the following functional group is present at C1 carbon of alpha D glucose in sucrose? Now at alpha D group of the sucrose, we are having this kind of positioning, right? Now at this position, I am having carbon which is bonded to oxygen over here as well, OR group over here, OR group over here. So when one carbon atom is bonded to 2 OR groups, this is actually called as the acetal group. So answer over here is going to be option D. This is acetal. If you will just look at the formula or structure of this particular group in sucrose, then you will observe this. Alright. Question number 68. Benzyl chloride is formed by treating toluene with chlorine. Benzyl chloride is formed in the presence of, now whenever we are going to form benzyl chloride, that is, this time the chlorination is basically done with the help of a free radical. Only then it is going to attack on the carbon atom which is outside the benzene ring, and now it is not attacking on the benzene ring directly. So this is done in the presence of light, because only in the presence of light free radicals can be formed. So answer is going to be option A. Okay. Question number 69. Monomer of Teflon is going to be tetrafluoroethylene. So answer is going to be option A. Directly, this is a direct question from polymers chapter. Next question 70. Which among the following is an essential amino acid? Now for this you have to learn the list of essential and non-essential amino acids. In this case, histidine is the essential amino acid. Proline, alanine and glutamine are considered as the non-essential amino acids which our body is capable of secreting itself. So answer is going to be option B here, histidine. Next question number 71. Tautomers of phenol can be which out of these? If I am talking about phenol here, let's say, now phenol is this. Now if I am going to make the tautomers of phenol, so for this I can also have this. Why? Because in this case, in the first position, I am having alpha hydrogens here. These alpha hydrogens will be removed and it is going to come here. I am going to get a double bond here and the OH bond will come here. So it's quite easy. So this can be a tautomer of phenol. Very nice. This cannot be the tautomer of phenol, or okay, wait. Okay, this can also be the tautomer of phenol. Why? Because over here as well, I am having hydrogen. Right? Now, when this hydrogen is liberated from here, this H plus will be moving here. This is going to come here. This bond will move here and this pi electron is going to come at this position. Okay? So, because of these, let's not make this more hodgepodge. Alright. So, over here, this is going to move here. Right? Simply because of this, now this bond is going to come here and ultimately the double bond will also be formed between these 2 carbon atoms, and this is also going to form a phenol. But at this position, in this particular in D option, we cannot have the tautomer of phenol. Why? Because in this case we are having sp2 hybridized CH bond. This carbon atom is sp2 hybridized, and the cleavage of CH bond which is in which the carbon atom is sp2 hybridized. That is quite difficult because that bond is quite strong. We cannot cleave it. Okay. So answer is going to be both A and B over here. Answer is option C in this case. Next question number 72. Which of the following is most basic? So most basic is asked. Now for most basic, the lone pair in which of these following is most easily donatable. That we have to see. Now this is a lone pair with on the sp3 hybridized nitrogen atom. So this is very easily donatable. That's fine. Now in the option B, now over here I am having methyl. Now methyl is actually providing the plus I group, which is intensifying the electron density of the lone pair, and now it is even more available for donation. So B is even more basic as compared to A. Okay. Now talking about C. Now C is also having a lone pair, but over here I am having oxygen, which is having a minus I effect. Sorry, this is having a minus I effect. That is, it is going to attract the electron density of the lone pair. Alright, now talking about the D option. Now in this D option, the lone pair is actually utilized in the resonance of this ring and making this ring more aromatic. So in the D is actually minimum basic. So if I'll just arrange them in the order, so D is least basic. Okay, D is least basic, then I'm going to place C, then A, and then B. So B is going to be most basic, so answer is going to be option B. Alright, question number 73. Which of the following polymers is formed via chain growth polymerization? Chain growth polymerization means we are going to have, these are going to show basically the addition polymerization, or alkene should be present. Only then the addition reaction will occur. So this is present over here in the case of polyvinyl chloride. So answer is going to be option A. Alright, question number 74. Select the correct statement in relation to natural rubber. Natural rubber over here is going to have the repeating units like this. Definitely. These are not cross-linked polymer. So this is not the answer. This is, okay. Now over here answer is going to, this is not a condensation polymer as well. Answer is going to be option C by default. This is a repeating unit over here is this. Next question number 75. On distilling sodium salicylate with soda lime, the product formed is what? Now if I'm having sodium salicylate, now what is sodium salicylate? This is sodium salicylate in the presence of soda lime. Now what is soda lime? Calcium hydroxide, CaO with NaOH, right? Now this is going to cause decarboxylation reaction. CO2 will be removed from here and I am going to get ultimately phenol out of it. So answer is going to be option C. Next question number 76. Select the correct match. Now over here the correct match is going to be which one of, incorrect match. Select the incorrect match is asked. Okay, cellulose is made up of beta-D-glucose, so this is a correct match here. Sucrose is going to have the glycosidic linkage between the glucose and the fructose unit. This is also correct. Lactose is actually made up of beta-D-galactose. Beta is there, beta-galactose, so this is an incorrect match. DNA is having a phosphodiester linkage. This is quite evident, so this is also right. Answer is going to be option C here. Alright, question number 77. Norethindrone and ethinyl estradiol are which of these drugs? Neurotransmitters, anti-fertility drugs, bactericidal or bacteriostatic. So this is going to be anti-fertility drugs. In NCERT it is mentioned in the examples as well. Please go through the NCERT. In each and every example you have to learn whatever is mentioned in the NCERT. Alright, so these are the anti-fertility drugs. Next question, number 78. Now these are the two compounds. These can be distinguished from each other with the help of which of the following tests? First is your iodoform test. Now if I'll just think about iodoform test is basically done where COCH3 unit is present. So in this case COCH3 somewhat is present. So in this case iodoform test will be positive. So this is going to give positive iodoform test. Alright. Now, but this is not going to give positive iodoform test. So, yes, using iodoform test, we can distinguish. Neutral FeCl3, definitely, because FeCl3 test is done for phenol and phenol unit is present in this case, but not in this case. So, positive neutral FeCl3 test will be given for this, and again, we can distinguish them. So, answer is going to be C, both A and B. Simple? Alright, question number 79. Which of the following nitrogenous bases is derived from purines? Now purines are basically of two different types, adenine and guanine. So just check, do we have adenine and guanine? So we have guanine here, answer is option D. Okay, pyrimidines are cytosine, uracil and thymine. So you all are well aware about this. Question number 80. Which of the following is not correct? Not correct is asked. Now over here orthonitrophenol is less acidic as compared to this one. So this is, okay, not correct is asked. Alright, so this is absolutely correct because over here there is going to be intramolecular hydrogen bonding will come. So because of this, the statement is absolutely true. Then chelation occurs in this because of the, again, same reason, intramolecular hydrogen bonding. So this is also right. Methyl salicylate is present in oil of wintergreen. This is also correct. Now talking about the fourth option, bond angle of ROR dash, which is an ether, is less than 109 degrees 28 minutes due to lone pair repulsions. No, this is wrong actually. It is more than. It should come over here more than, because of the repulsions between the alkyl groups R and R prime. Because of the repulsions, the bond angle increases and it can be nearly 111 around. So bond angle is not less, it is greater than that. Okay, question number 81. Which of the following compounds is responsible for the transmission of hereditary characteristics? That is by default a DNA. So everybody knows that. This was a bonus question for all of you. Answer is option B. Question number 82. Glucose does not react with which of the following cases? Glucose actually reacts with all of these three. It does not react with Schiff's reagent. Now what is the Schiff's reagent? Where we are having carbon-nitrogen double bond, where the nitrogen is also bonded to two alkyl groups. So over here this is not going to show any reaction with the Schiff's base. Alright, question number 83. A, B and C in the following is going to be which one of these? Now these are all different reagents here. All are some sort of reducing agents only. So if I'll just talk about the first one. Over here I'm having an hydrazine in the presence of ethylene glycol. This is glycol here. This is L. Alright, this is ethylene glycol in basic medium. For basic medium we usually use KOH. Now this is basically used to remove or to reduce the carbonyl group. Carbonyl group is actually converted to the hydrocarbon. So this is removed over here and I am going to get this ring, these two rings with double bond. So over here this is formed basically. In A this is formed. Alright. In the case of B, ranitidine, nickel and hydrogen. Again this is a strong reducing agent. Everywhere everywhere hydrogenation is done and I am going to get hydrogens here added and over here also added. So I am going to get this as the product. And for C, NaBH4 and in methanol, I cannot reduce the double bond here. Only this is reduced to alcoholic group. So this is also the answer. Answer is going to be option D here. Alright. Question number 84. Which of the following, what is the correct statement about birth control pills? Correct statement. Please highlight. Correct statement is asked. It contains estrogen only? No, not necessarily. It contains progesterone? No. It contains a mixture of estrogen and progesterone derivatives. That is a correct statement for sure. Progesterone enhances ovulation? No. So answer is option C here. It contains a mixture of estrogen and progesterone derivatives. In the anti-fertility drugs or birth control pills, we are having the mixture of these. Alright. Question number 85. When cyclohexanone is treated with perbenzoic acid. Now what is a cyclohexanone ring? Cyclohexanone is this. Now when this is treated with perbenzoic acid. Now perbenzoic acid is basically responsible for the formation of lactam. Okay, what is lactam? Where we are going to have at the alpha position, oxygen is inserted between the carbonyl carbon bond. Okay, then the product formed over here. Now earlier we were having 6-membered ring. Now we are going to have 7-membered ring with 1 member as an oxygen atom here. Alright, so answer is going to be option B in this case. This is a lactam. Okay, question number 86. Reactivity order given alkyl halides towards E2 mechanism is going to be. Now whenever we talk about elimination reaction, tertiary is the most reactive alkyl halide, then secondary and then primary. Now over here we are going to follow the same trend. This is secondary, this is primary and this is also primary. But in primary as well, where the where we are having sterically hindered environment, there the reactivity will by default decrease. So maximum reactivity will be for secondary. This is primary, this is also primary. Maximum reactivity will be for B, and this is present actually in just one option. Then for C, because this is sterically less hindered and this is sterically more hindered, then last reactivity is for A. So answer is going to be option C. Question number 87. Find the correct match here. Now, which is the correct match? Pteryline is a copolymer of terephthalic acid and ethylene glycol. Absolutely correct. Buna-N is a copolymer of acrylonitrile and buta-1,3-diene. Absolutely correct. These are the monomer units here. Neoprene is a polymer of chloroprene. That is also correct. So all of these are correct. Answer is option D here. Question number 88. Now over here I am having a nitrogen atom with a positive charge, which is making a 4 bonds here when it is heated. Now this is basically cope elimination or cope reaction, where whenever we are having a quaternary nitrogen atom, this tends to undergo elimination of the small group. Okay, the alkene formed as a major product in the above elimination reaction. This is cope elimination reaction. Please write down. The name of the reaction is cope elimination, where the smaller alkene tends to move out. So over here, methyl cannot form any alkene. Only 2 of the groups can form alkene. One is the butyl and another is the ethyl group. Now ethyl is smaller group as compared to butyl. So ethyl will be removed and this is going to form alkyne over here, and I am going to get this as my answer, option B. Alkene is asked to me, so answer is option B. Okay, question number 89. In the presence of peroxide, hydrogen chloride and hydrogen iodide do not give anti-Markovnikov product. Now whenever we do the addition reactions or Markovnikov or anti-Markovnikov reactions, when we do, HBr in the presence of peroxide only gives the anti-Markovnikov product. Otherwise HCl and HBr do not do that. Why is that so? Because HCl is actually having stronger bond as compared to HBr. So if I want to break this bond, or if I want to cleave this bond between H and Cl, this requires more energy. So this step over here is going to be more endothermic in the case of when I want to cleave the HCl bond. Okay, so this is the case of HCl. Now talking about HI. Now HI, cleavage of HI is very easy. It is easily cleaved. But when I am going to add iodine into the alkene, that particular reaction is endothermic there. When that particular reaction is endothermic, that means I need to give energy. So if I need to give energy, that means that reaction becomes more difficult there. So in this case, cleavage is more difficult. So cleavage is the endothermic step. But in this case, addition of iodine to the alkene ring, to the alkene group or to the alkene molecule is an endothermic reaction. So over here the answer is going to be one of these steps is endothermic in both the cases. Although the steps are different, but one of these steps is going to be endothermic, and we need energy to make it an anti-Markovnikov product. That is why they do not show anti-Markovnikov or the formation of the free radicals. So answer is option C here. Okay, question number 90. In which of the following polymers ethylene glycol is one of the monomer units? Now what is ethylene glycol? CH2OH. CH2OH. Now whenever we are going to have this as the monomer unit, this H and H are basically removed. Now let's see over here in which of the case do we have that. OCH2, okay, in this case we are having this over here. In any other case we are not having anything like that, so answer is going to be option A. Okay, question number 91. Consider the following reaction over here. Now nitrobenzene in the presence of tin and HCl. Now this is a reducing agent, it is going to form aniline. So I am going to get over here aniline, NH2. Okay. Now when this is getting brominated in the presence of bromine water, that means now this, first of all, this ring is highly activated. Okay, because of the plus R group. Moreover, in the presence of water as a solvent, more number of electrophiles, Br positive, will be formed, and hence the attack at the ortho as well as at the para position of aniline is quite easy, and I am going to get all tri-substituted product, tri-bromo-aniline. So answer is going to be option D here. If instead of water as a solvent, I would have any other non-polar solvent, like for example, bromine with CS2, then I would have got mono-substituted product. But because of water, tri-substituted product is formed here. Okay. Question number 92. In the reaction where I'm having this type of epoxides, in the presence of hydrolysis, acidic hydrolysis I should say, A is formed and A is your product. So what is A basically? What is the structure of A? So whenever we are having an acidic medium, this H+. Now over here this is a lone pair. This lone pair is going to attack on this H plus and ultimately this bond will break. Okay, and now the lone pair of water, the lone pair of oxygen from water is going to attack at this carbon atom, and I am going to get the OH at this position, which is coming from the water, and this oxygen will move on the terminal carbon atom as such, and I am going to get answer as option A. Question number 93. Which of the following acids is a vitamin? Now, which of these are vitamins? So, over here, ascorbic acid is a vitamin. Otherwise, all are or not vitamins here. Ascorbic acid is a vitamin. Answer is option B. Question number 94. Which of the following compounds additional resonance with the ring is not possible? Please remember, it's asked additional resonance is not possible. So, let's see over here. Now, I am over here having NHCO. Now, instead of despite the presence of a ring, I am having over here this group. So, extended resonance is coming from this group, NHCO group. Carbonyl resonance is, conjugation is maintained till the carbonyl part. Then over here we are having again the lone pair. So, this is also responsible for the resonance apart from the benzene ring. This is also having resonance, CONH2 group with the lone pair and the carbonyl CO group. Over here, NH3 positive, this is not going to show any extended resonance because NH3 positive is going to discard all the resonance present over here. In this case, only in the benzene ring we are going to have some sort of resonance. Alright, next question number 95. Which of the following B group vitamins can be stored in our body? Which of these can be stored in our body? 1, 2 and 6. They cannot be stored in our body. B12 can be stored easily in our body because it is solubilized. So, answer is option D. Question number 96. A simple method to remove peroxides from ether is to treat them with an aqueous solution of which of these? Ki, KCNS, Na2S2O3, Br2. Now, over here, peroxides are basically some sort of oxidizing agents. Now, if I will be treating this with I negative, now I negative will be easily converted to I2 and peroxides will be reduced. Okay, peroxides are reduced and ultimately removed from the reaction mixture, and iodide are basically oxidized to iodine. All others cannot be furthermore oxidized over here, so answer is going to be option A, I negative. Alright. Question number 97. Which of the following containing carbonyl group will give crystalline compound with this particular compound? Now, what is this particular compound? This is 2,4-dinitrophenyl which is 2,4-DNP, which is going to show some sort of characteristic, or this is the test to show the carbonyl groups. Ketones and aldehydes show a positive test for this. So, over here we have to find out ketone or aldehyde only. Now, this is your acid chloride, so this is not going to show. This is a ketone, so this is going to show a positive test. This is an ester, will not show. This is an amide, this is also not going to show. So, answer is going to be option B. Okay. Question number 98. Nitrobenzene on reduction in the presence of lithium aluminium hydride gives what? Now again this is undergoing reduction. Nitrobenzene is going to directly form aniline, easy one. So answer is going to be option A. Question number 99. Which of the following statements is correct? Now they are asking you to mark the correct statement. Some tranquilizers function by inhibiting the enzymes which catalyze the degradation of noradrenaline. Absolutely correct. Tranquilizers are basically used for the patients which are in depression. So these are some sort of antidepressants. So they are basically going to degrade the noradrenaline secretion. So answer is option A only. Tranquilizers are narcotic drugs? No. Tranquilizers are chemical compounds that do not affect the message transfer from nerve to receptor. That is wrong statement. It does affect that because it is an antidepressant. Okay, now tranquilizers are chemical compounds that can relieve pain and fever? No, because they are antidepressants. So answer is going to be option A. Correct statement is option A here. Question number 100, last question for chemistry portion. In carbylamine test, the reaction intermediate formed is what? Now this reaction is done basically in the presence of chloroform CHCl3 along with alcoholic KOH. Right, this is done. So alcoholic KOH causes elimination from here. HCl is removed out and I am going to get CCl2 carbene here. So I'm going to get carbene as an intermediate here. So answer is going to be option D. Carbene as an intermediate is basically used when we are having Riemann-Tiemann reaction as well as carbylamine reaction. For both these reactions, carbene is the reaction intermediate. All right. So with this, the chemistry discussion portion is actually ended. Now further on, we will be having a discussion of physics portion. Thank you so much, everyone.
Hello everyone, welcome to NEET Extramarks lecture series, and today, as you know, we are discussing minor test 4, which was conducted just day before yesterday. And what I'm going to do is we are going to discuss physics paper, and I'm sure all the other paper that have been discussed that have been clear to you. So let's begin. Before guys we begin the physics part, let's very quickly discuss the this lecture series, this NEET AIRTS, that is All India Rankers test series as we are calling it. So guys, rankers, that is included in the name, and that is also the purpose of the test series, is to help you improve your rank in the actual NEET exam. So all these papers that are being conducted, that are based on we are trying to predict what kind of question will be coming in the NEET exam. So the paper pattern, the structure and the types of question that are there, so that basically we are trying to replicate the NEET exam. So it will, this test series will really help you in the NEET exam. Not just this year, this has been happening in the previous years also, that these AIRTS students who do good in AIRTS, they generally do very well in the actual NEET exam also. So before we begin, let's very quickly look at the basics. So it started, although it on 28th November, but so far 4 tests have been conducted, which have been minor tests. What is minor test? That also I'll tell you. Classes, of course, these are for students who are giving the NEET this year in 2023, and subjects, of course, based on NEET: physics, chemistry, botany, and zoology. So number of tests, so guys, we will have 4 minor tests. So this was the actually last minor test that we had. So this was based on the partial syllabus. Now we are having 2 mega tests which are based on half syllabus. So 2 mega tests, one for 11 syllabus and one for class 12 syllabus. And then we have 6 full syllabus, exactly NEET based pattern exams. So that will be after the 2 mega tests. Test pattern, of course, same as the NEET pattern. And then test duration, again same, language is English. And just to give you the schedule of the NEET Extramarks AIRTS, so minor test 4, that was on 9th of January, and then next we are having a mega test 1, that is after 2 weeks. So mega test 1 and 2, as I told you, this will be the half syllabus for 11th and 12th, and then we'll having the full test, okay, full syllabus, both 11, 12, everything. So these will be exactly NEET based papers. Alright guys, so this is mostly it about the Extramarks AIRTS, and now let's continue with our paper discussion. So as I told you, I'll be discussing the physics paper. So we'll go through very quickly, we'll go through all the question. Of course, I'll tell you only the solution because we won't have the time to discuss the actual concept. I won't be able to teach you the concepts behind, but through the solution only I'll try to describe you what is the concept and how do we tackle such questions in general. Alright guys, so let's look at the first question. So this is a question from optics. Okay, it is only from reflection, and this question is asking what will be the length of the mirror, what will be the minimum size of the mirror that is fixed to the wall in front of him. Alright, so if you draw the diagram, so basically there are 2 walls here which are what, 3 meter high. So both the walls, they are 3 meter high. Okay, let's say this is the floor. Okay, and in between them the person is standing. What is the height of this? 1.6. So little bit taller than half of the wall. So this is the person, let's say. Alright, so there is one mirror on one of the wall in front. So let's say on this wall we have this mirror. Alright, so this is our mirror. So this person, what he wants to do is he wants to see the wall which is behind him using this mirror. So how will that happen? So and what will be the minimum size of the mirror? So that is the question. So how will that happen? So basically guys, he will be able to see the wall, of course, when the light rays from the wall will strike the mirror, and then they will reach the person's eyes. So we can assume here that eyes are like directly at the highest point, at its maximum height. Although it is not exactly true, my height is not exactly at my head, but the difference is so less that we can assume that the eyes are on his head. Even if we don't assume, even if we consider it slightly below, won't really affect the answer. So yeah, let's say the eyes is here. So now if he wants to see the object, the wall behind him, let's say, so how will the light reflect? So he should be able to see the top point of the wall. So how will the light reflect? So light will go towards the mirror. Okay, it will get reflected and then it will reach the person's eyes, right? That is going to be how the reflection will be. So this mirror should be able to reflect that light which is coming from the top. So that is the first thing. Second, he should be able to see the bottom of the wall also. So the light from the bottom should strike the mirror. Okay, and then after striking the mirror, it should reach the person's eyes. So that is going to be the reflection from the bottom of the wall. So guys, that is the diagram, and of course so the light from the top of this wall should strike the top of the mirror. Okay, because if the mirror is above it, then that mirror is useless. We don't need that part. We only need that part which is the light from this top of the wall is striking the top of the mirror. So this is the top part of the mirror, and then similarly at the bottom, light from the bottom will strike the bottom part of the mirror. So this is the bottom of the mirror. So now we got it that if we talk about the mirror, so the mirror is basically between this. So this is the height of the mirror. So now it is just basically geometry and the fact that angle of incidence is same as the angle of reflection. So here we know the height of this person, which is 1.6. We know the total height. So right, so these two angles, they are equal, and using the concept of similar triangle, we can indeed find what is this distance. Okay, what is basically from the wall, what is this distance? So total is 3, this is 1.6, so the remaining is 1.4. So the remaining 1.4, so this will, half of that will be 0.7 and half of that will be 0.7. So the mirror basically you can easily find what will, where will be the mirror start using the property of similar triangle. One triangle basically we can take this one. So if I connect, so this is the normal. So one triangle we can take the larger one, and another one we can take the smaller one. So if I name it A, B, let's say C, and this is D and E. So two triangle ABC and CDE. These are two similar triangle because one of the angle is 90. So all three angles are equal. This is angle incidence reflection. So using similar triangle, we can find what is going to be DE. So guys, DE is going to be the half of AB, right? So that is how we can find D. Total, if we half of, so AB plus DE combined, it is 1.4. So using that we can find D. D will be 1.4 by 3. Similarly, at the bottom also we can do similar thing. We can draw the normal here. Alright, and using the normal we can use the property of similar triangle. Basically one of the triangle will be this, and the other triangle, if I draw the normal, so the other triangle will be at this point, let's say. So this is F, G and H. So F, G, H, that is one triangle, and then this smaller triangle, just like we have taken C, D, E. So we have to take the smaller triangle here also, and that we can take, let's say, this point we can take X and Y. So X, Y and this H point. So this will be the another similar triangle. So using that we can easily find. So that is just geometry. We are not doing it here. That will you get a little bit of calculation, but this is the idea, that light from the top of this wall will strike the top of the mirror, reach his eyes. Light from the bottom will strike the bottom of the mirror and reach his eyes. Alright, so that is what you have to do in such question. Draw the diagram, and that will give you the idea. Alright guys, let's continue. So if you solve this, the answer you'll get is 1 meter. Alright, and note that this this won't depend on actually the height of the person. So even if this person was differently, if instead of 1.6 he was 1.7, even then you'll get the same answer. Alright guys, let's continue. Convex mirror. So again, this is from optics, image of a real object, mark the wrong statement. So basically we are using convex mirror. So you need to know what kind of images are formed by the convex mirror. So guys, the convex mirror, they do not form real images. Alright, so real images they do not form. So that is the answer, because question asking the wrong statement. So wrong convex mirror, they only form virtual images, right? Virtual, and they form diminished images. So that is right, all three, virtual, erect and diminished. So ABC, all three are fine, but D is incorrect. So that is the answer. Alright. Next one, object placed in front of a concave mirror, focal length is given, and it produces a virtual image. What is the position of the object? So it is just what they are asking us is the mirror formula, right? 1 by V minus 1 by V plus 1 by U. Okay, let me write it here. 1 by V plus 1 by U equals to 1 by F. So focal length is given. Of course, concave, so focal length we have to take negative. Focal length is given, and here they are not giving us the V directly, but what they are saying is that the height is twice. So we know that the height formula, which is height of image divided by the height of object, and that is minus V upon U, right? And that is twice. So that is given here. So this ratio we can use, minus V by U equals to 2. So I can write that V equals to minus 2U. So here I can replace V with minus 2U. So V 1 by minus 2U plus 1 by U equals to 1 by F. This is the equation I'll get. So now you can solve it and get the value of U, which is the question. F is given. So this is just calculation, right? Okay, this should be plus minus 1 by 2U plus 1 by U. So that is 1 by 2U equals to 1 by F. So this you can easily find U from here, which will come out to be minus 7. Of course, minus, guys, U is always negative. So that is why, of course, B cannot be the answer. U must be negative only. So that is why the answer here is coming out to be C. Alright, let's continue. When light wave. Okay, so we are refracting. So this is about refraction from rarer to denser. So guys, what happens when the light ray goes from rarer to denser? So denser, we know that the speed is less. That is the meaning of denser medium, that the speed of light is less. So now they are asking about speed and wavelength, all that. So guys, the speed decreases, right? It is going from rarer to denser. So the speed is going to be less. So the speed is decreasing. So A and C are of course eliminated. Now what about the wavelength? So guys, frequency won't change. That we should know. Frequency won't change. So if I write the formula of speed, that is wavelength into the frequency. So this is decreasing. So the wavelength must also decrease, because this is constant. So if velocity is decreasing, then wavelength must also decrease if frequency is constant, so wavelength is also decreasing. That is why this is the right answer. Okay, next one. So this is about so length of the vertical. So there is a pole in the water lake. Okay, underwater. So fish is underwater. So what will be the length of the pole? So this is basically about apparent depth and apparent height. So basically, if you draw the diagram, so this is the water surface, let's say, and this is the fish is just below the surface, and this is the pole. Okay, what is the length of the pole? 24 centimeter. But so according to this fish, what will be the length of the pole? That is the question. So this is apparent height, which is the opposite of apparent depth. According to the fish, what is the apparent height of the pole? That is the question. So now we can use the formula of apparent height. And guys, what is the formula of apparent height? So that is apparent height is equals to actual height divided by the refractive index of the medium. So generally apparent height will be more, just like apparent depth, it is less. So what is apparent depth? Apparent depth is the actual depth divided by the refractive index. So here the apparent height to the fish, that will be the actual height divided. So here the refractive index must be multiplied, because apparent height will be more, right? It is kind of the opposite. So apparent depth is less and apparent height is more. So to the fish, it will appear as if the pole is taller, right? So here it will be the height into the refractive index of the medium in which the fish is there, because outside it is 1, otherwise we have to divide by the outside also, but outside is 1, so yeah, it is just height into the refractive index. So height is 24 into refractive index 4/3. If we do that, the answer we get is 32 centimeters. So here B actually should be the answer, 32 centimeter. All right. Next one, ray of light strikes plate, glass plate, reflected and refracted rays are perpendicular. What is the refractive index of the glass? So that is, so if we draw the diagram, that will give us the idea what is happening. So guys, this is the let's say surface of the glass plate. So light is coming from air. Okay, so let's say this is the glass, this is the air. So light is coming on this plate, it strikes. Okay, and it reflects also and refracts also. Both things are happening here. So it reflects. What is the angle of incidence? 60 degrees. So that is given that this angle is 60. Okay, and now what is going to happen is refraction is also happening. So if I draw the refraction, so this is the refraction. Okay, let's bend it slightly more, because currently it is looking like straight only. So let's bend it a little bit. So yeah, this is the refraction. Okay, and these two are perpendicular, that is given. So if I draw the angles here, so this is of course 60, because this is a reflected ray. So if this is 60, this must be 30, and if this is 30, then because these two are perpendicular, so this must be 60, and if this is 60, then this must be 30. So this is how we got the angle of refraction. So now we know angle of incidence, we know angle of refraction. Snell's law, mu1 sine I equals to mu2 sine R. That is your answer. So if we try to solve it, so mu1, what is mu1 is 1 into sine I, which is sine 60, equals to mu2, which is the refractive index of glass. That is the question. Find the refractive index of glass. Let's say it is mu, into sine R. So guys, sine R, what is sine R? Sine 30, right? So sine 30 is of course 1/2, sine 60 is root 3/2. If you put the values 2 and 2 will cancel, so mu will be root 3. So root 3, that is option A, so that will be the answer. Alright guys, let's continue. Two thin lenses, focal lengths are given. What is the combination of what is the focal length of the combination? That is the question. So whenever guys we have two lenses which are in contact with each other, what we have to do is we can add the power, or what we can write is net power, okay, equals to P1 plus P2. That is the net power. So now if we add the power, so power is of course 1/f. So here this is plus 20 focal length, this is minus 20. So 1/f plus 1/f1 plus 1/f2. So 1/f1 plus, so 1/f2 is actually minus f1. So because focal length are 20 and minus 20, so they will cancel each other, and the power net is 0. And guys, power is 1/f, so if power is 0, focal length is 1/0, which is infinite. So that is why the answer here is infinite, because the power actually net power becomes 0. Alright guys, let's go to the next one. So now this is again, so okay, this is a plano-convex lens. So one side is plane, other is the convex. Radius of curvature of convex is given, focal length of the lens is given, what is the refractive index? So what is this question? Guys, the first thing that should come to your mind whenever you see a question is, what is the topic? What is the formula which we need to apply? So this question is directly from Lensmaker formula. So we should know what is the Lensmaker formula. So if I write the Lensmaker formula, that is mu minus 1 by r1 plus 1 by r2, right? That is what? That is your 1 by f of the lens. So here guys, mu is they are asking, and r2, so one of the surface is planar. So for a plane surface, the radius is infinite, so we can cancel the r2, 1 by r2, which is because if r2 is infinite, so 1 by r2 is 0, so 1 by r2 is going to cancel. So mu minus 1 into 1 by r. So everything is given, we just need to put the values and we will get the value of mu. So what is the focal length, guys? Focal length is 30, so 1 by 30 equals to 1 by f. So 1 by f is 1 by 30, mu minus 1. Okay, 1 by r1. So guys, what is 1 by r1? So this is a convex surface, so we can take r1 as positive, so 1 by 10. That's it. So if we solve this, we'll get the value. Mu minus 1 is 1 by 3, so mu will be 1 plus 1 by 3, that will be 1.33. Alright, so simple question from Lensmaker formula. Okay, next one. So now again it is a question from Lensmaker formula. So this, if we talk about this one, so guys, what is the okay, let me write the formula again, Lensmaker formula. So actually the formula is 1 by f equals to mu r. So this we have written mu minus 1 because outside it is air, but the actual formula is mu r, that is mu of the lens divided by mu of the surrounding. Okay, so mu lens divided by mu of the surrounding, let's say mu s, minus 1, okay, 1 by r1 plus 1 by r2. So this is the like actual Lensmaker formula. So here let's see, initially it is refractive index is 1.6, power is 1, power is 5. So if power is 5, power is what? 1 by f, so this is 5. So now if we write the equations here, initial what is given, 5 equals to mu L by mu s minus 1. So initially it is in air and refractive index is mu L is 1.5. So 1.5 minus 1 and this thing 1 by r1 plus 1 by r2. Right, so 1.5 minus 1 is 0.5, goes to denominator, so 5 divided by 0.5 becomes 10. So 1 by r1 plus 1 by r2, this whole thing is 10. So this I can write 10 equals to 1 by r1 plus 1 by r2. Right, that's it. Let's say this is the first equation, and second we can write, because finally it is placed in a liquid of refractive index mu, and the power becomes, focal length becomes 100 centimeter. So 1 by f will be, focal length is 1 meter, so power basically becomes 1 diopter. So power is 1 equals to it acts as a divergent. So power we have to take negative, because divergent lens means it is like basically concave lens. So concave, we know that the power we have to take negative, right? So this we have to take minus 1. That was the important thing, because that is why they are saying divergent, because divergent means the focal length is negative, right? It is diverging rays, it is not converging. So this is minus 1, that is 1 by f. Okay, mu L, which is what we know, 1.5 refractive index of the lens, divided by mu of the liquid, now it is in the liquid, minus 1, and again same thing, 1 by r1 plus 1 by r2. So this is the equation, guys. So 1 by r1 plus 1 by r2 is 10. We know the value from the first equation. So minus 1 equals to 1.5 divided by mu minus 1 into 10. So this is your equation. Of mu is the only variable, so this you can solve. I am sure you can solve it easily if you do that. So you will get the answer, that is 5 by 3, right? Because 10, if you go to this side, this will be minus 0.1, and 1 by 5, so this will be 1.1. So mu will be 1.5 divided by 0.9. So 1.5 by 0.9, that is going to be 5 by 3. That is will be your answer. Okay. Next one. 10th one. What angle will a light incident on equatorial plane so that emergent ray may gaze the second surface of the prism? So this is basically, guys, about the okay, mu is also given. So this is about the prism. We should know that in the prism, so basically they refract light, and there is a this is equilateral. So we should know the emergent. So this is, they are talking about the emergent ray which is grazing. So basically we can say that the emergent angle here, it is just grazing, so emergent angle with the normal will be 90. So the what we should remember is one of the formula, prism, which is a generalized formula which is valid for the prism. Let me write it with a different color. So that is A plus del equals to I plus E, angle of incidence plus angle of emergence. So that is your formula of minimum deviation. So that is what we can apply here to get the value, which will going to be the so they are talking about the incident angle. So here incident and emergent both are same, both are 90. So emergent is 90 and incident is also 90. Alright guys, let's go to the next one. So now they are okay, this is a little bit complicated one because here the refractive indexes are different on the different sides. So this is the air, and this is mu3 is 4/3, and the lens is refractive index 1.5. So what is the focal length of this whole system? So actually we cannot use lens maker formula directly because lens maker formula is for a like single surrounding. Here the surroundings are different on left and right side, so we cannot do that. So what we have to do is we have to use the formula of refraction on the curved surfaces, because actually lens is what? Lens is refraction happening twice, so on the left surface and then on the right surface. So we need to do that and find the focal length that way. So what is the formula of refraction on a curved surface? So that is, if I write the formula, mu2 by v minus mu1 by u, this is u, u equals to mu2 by v minus mu1 by u, minus mu1 by u, that is mu2 minus mu1 by r, right? That is the formula of the refraction on a curved surface. So first we need to apply this formula on this surface where the r will be positive, okay, because this is a convex surface, the first one, so r is positive. And what is mu1? Mu1 is the other medium, the next medium. So mu2 in the first case will be 1.5. So now if I write the 1.5 divided by v minus mu1 is air divided by u. So guys, they are asking the focal length. So focal length basically means when the object is at infinity, where is the image? So we can take u as infinity because the object is at infinity, right? That is the meaning of focal length. But of course this we cannot take focal length v because this is not the final image. Final image will be formed after the refraction from the second surface. So when I write the equation for second surface, there the v will become equal to the focal length. Alright, so let's see. So mu2 by v minus mu1 by u is infinity equals to mu2 minus u1, which will be 0.5 divided by r, which is going to be 20. So this is your equation. This is 0, of course, so 1.5 by v equals to 1 by 40. So here we'll get the v. V is what? V is 40 into 1.5, which is 60. So this is your first image, but this now image will act as the object for the second surface. So now I have to do the formula again. So now the formula will be mu3 by v. So now the final image is my focal length, because when the object is in infinity, image is formed at the focus. So mu3 by f minus now the okay, this should be mu2 actually, because it is for the lens. So mu3 by f minus mu2, because initial here is the lens, divided by u. So here u is same as v, because this is your now this is acting as the object, is equals to mu3 minus mu2 divided by r. R is going to be minus 20 because now this is a convex surface. Alright, so that is why the r here we have to take minus 20. So now if you put the values, everything is given. V 60 we calculated, mu3 mu2 4 by 3 1.5. If you put all the values, you'll get the value of f, and if you do the calculation, you should get 40 centimeter. It's a little bit of complicated one because we have to apply the refraction at two surfaces first, and this one and this one. So in this type of question you have to do this only. There is no shortcut here because it is basically refraction happening twice. Okay, next one. So again, this is from a prism. Angle of prism is 30 degree. Light falling at angle of incidence is 60. It is given, and it basically retrace the path after the reflection. So here this other surface of the prism is made reflected. Okay, it is made silvered surface. So now there is reflection. So of course we know reflection, it retrace the path when it is the angle of incidence is 0, then angle of reflection is also 0. So in that case it will retrace the path. So basically it is perpendicular to this surface. So that is why all the diagram is not slightly incorrect, because this image is basically perpendicular to the mirror. That is what they are saying. So if this is 30, this is perpendicular, so this must be 60 degree, right? This angle is 60 degree because total must be 180, so 30, 90, so the remaining is 60. Okay. Now, this is of course the normal to the first one. So if this is 60, what is this angle? Let me draw the angle for you. What is this angle? This one. So guys, this one of course, because total is 90, this is the normal here. So this is 60, so this must be 30 degree. So this is what? This is nothing but your angle of refraction, which is 30. So angle of incidence is 60, angle of refraction is 30. So now you can easily use Snell's law to find the value, which is going to be √3, which is 1.73 approximately, right? So Snell's law. We just need, using the diagram, we just needed to find the angle of refraction, and now it is just Snell's law. All right. Okay, this one. So these type of questions are also common. Fish. What is the radius of the? How much will the fish able to see? Fish will not be able to see at everywhere, because let's say this is the surface of water. If there is a light ray which goes like this from the eyes of the fish, it will actually reflect, because TIR, total internal reflection. So these rays, these are the like extreme rays to which fish can see. So these happen at critical angle. So actually the C which is written, these are critical angle, right? So here instead of C we can also write IC. So IC, this is the critical angle, and we know the formula of IC, right? So sin IC here, which is mu2 by mu1. Mu2 is 1, outside is air, divided by the refractive index of water, okay, which is what, 4 by 3. So this is 1 by 4 by 3, so 3 by 4. So sin IC, we know guys, it is 3 by 4 here. So 3 by 4. Now the question is to find R. We know the H also. So guys, if we know H, we know sin. So what we can do is we can use tan theta here. So tan theta will be R by H. There is one more way, we can use cot theta. Okay, why we are using cot theta, we'll see. So cot IC is, so tan is perpendicular upon base, cot is base upon perpendicular, so H by R, right? And we know the formula, that is cot square theta, cot square IC plus 1, that is cosec square theta, right? Cosec square IC. So now because we know the value of cosec, cosec is 1 by sine, so sine is 3 by 4, so cosec must be 4 by 3. So now we know everything. Cot is H by R, so H square by R square plus 1 equals to cosec, which is 4 by 3 square, so 25 by 16 by 9. So this is 16 by 9. So minus 1, it will become 7 by 9. So H square by R square is 7 by 9. So H, we know, if we know the value of H also, which is 12 square, so from here we can find easily the R square, and then we can take under root and we'll get the value of R. So if you do this, a simple calculation, put the value of H, you will get the value of C, which is this 36 by √7 centimeter. All right. So yeah, this is basically from, we need to use the concept of TIR and critical angle. Okay. Next one, again this is from critical angle. So this is simple. Ray of light is incident, separating glass and water. Glass is refractive index 3 by 2, and for water it is 4 by 3. The critical angle of glass boundary. So so what is the critical angle? So guys, sin IC, we know it is mu2 by mu1. So mu2, what is mu2 here? The other one, which is 4 by 3, divided by the glass, which is 3 by 2. So this we get 8 by 9. So sin inverse 8 by 9, that is going to be our answer. So this was simple. Okay, two waves intensity ratio of maximum to minimum intensity. This is again simple if we know the formula directly for maximum intensity. So guys, if the intensity is I1, so the max intensity, okay, let's say I max. Okay, the I max is given by this formula. I max is root I1 plus root I2 whole square. That is I max, and I minimum is root I1 minus root I2 whole square, because actually it is the root I1 is what? Amplitude. So here we are adding the amplitude and then squaring it, which is the net amplitude, and then here also we are finding the net amplitude, but it is destructive. So net amplitude is A1 minus A2, and then for intensity we do the whole square. So now I1 we can take 9, I2 we can take 1, although it is the ratio, but we can put it directly because the ratio, so this we can take 1 and I2. Basically what they are saying is I1 and I2, so one is 9, other is 1. So I1 we can take 9 and I2 we can take 1. So root of 9 will be 3, 3 plus 1, 4, 4 square 16. So I max will be 16, and if we talk about I minimum, so 3 minus 1, 2, 2 square 4. So this is going to be 4. So 16 by 4, that is going to be our answer. 16 by 4 is 4:1. So that is the formula, you can remember directly. Alright guys, next one, fringe. Okay, it is from YDSC, young double slit. So they are talking about the fringe width. What is the formula of fringe width, guys? You need to remember this, d lambda by d. This is the fringe width. Alright, so we want to increase it. So we want to increase this thing, fringe width. So what should we decrease? That is the question. What should we decrease so that this increases? So of course, what is in the denominator that we should decrease, so the whole value will increase. So small d. What is small d? So small d is of course the separation of the slits, right? That is the small d. Capital D is the distance between the source, and lambda is of course the wavelength. So if we decrease small d, the whole value will increase, and we'll get the fringe width will also increase. So the answer is decrease small d. It means decrease the separation of the slits. Alright guys, next one. Okay, so what they are saying is, so it is again YDSC. So guys, YDSC, I am sure you know the diagram. So these are the two slits, okay, and this is my wall. So there is the interferences happening. Alright, so we know here this is the central maxima, and then we have minima, maxima, like that, bright, dark, bright, dark. So here what they are saying is, if we one of the two slit is painted, so one of the two slit is painted so that it basically transmit only half the intensity. So this is one first of the source is common, but first of the from the slit the intensity becomes half. So earlier the intensity was same. So if this is, let's say, I by 2 and this is I. So here guys, what is going to happen, the constructive and destructive interference will happen, but for the destructive interference they won't cancel it exactly, because the amplitudes are not same. So they because intensities are different. Amplitude is what, under root of intensity, so they are not same, they won't be able to cancel it. So that is why dark fringes won't be completely dark. Yes, there will be destructive interference, but the amplitude, net amplitude and net intensity will not be zero for the dark fringes. So that is why the dark fringes will appear more bright. Okay, first thing. Now what about the bright fringes? So guys, again, bright fringes, the net intensity will become less than the earlier intensity, because we are decreasing the intensity of one of the sources, right? So if we this basically we can use this also. I max is root I1 plus root I2 whole square. So earlier the I1 I2 both are same, but now let's say I2 becomes half, so I max will also decrease, right? And if you talk about I minimum, earlier they were cancelled, I minimum was 0, but now they won't cancel because I2 becomes I2 by 2, so they won't cancel. So I minimum will increase, I max will decrease. Alright, so if you look at the option, bright fringes will become less bright, and the dark fringes will become they will become less dark, so they will also become little bit bright. So that is why the answer is C. Dark fringes would be less dark and bright fringes would be less bright, because I max is decreasing and I minimum is increasing. Alright, next one, YDSC again. So guys, this is simple one. They are talking about the one of the this bright fringe coinciding with this, all that. So what we need to do in such question is, so n for n plus 1 of the blue light. So where is the n plus 1 fringe of the blue light? So guys, that is n d lambda by d. So for the blue light it is n plus 1, so n plus 1 d lambda for the blue one divided by d. Okay, this is coinciding with, so the gap is same as n for the red one. So n for the red one, d lambda, of course we have to take for the red, divided by d. So this is your this is the equation. So d, small d will cancel, capital D will cancel. Finally the equation you will get is n plus 1 lambda blue equals to n into lambda red. That is the equation here. right? So lambdas are given, okay, and you can find n, so not very difficult, right? And lambda blue is what, 7.8. No, that is lambda red, and lambda blue is 5.2 into whatever, so the power will also cancel. So you can easily find n using this equation. If you do that, you'll get the answer, which is going to be n equals to 2. Alright guys, next one. Okay, again from IDSC, intensity at distance beta by 4. Beta is the fringe width. So guys, if we talk about YDSC, if we I hope you know the pattern. So let's say this is the bright fringe, okay. So at the bright fringe, and this is the dark fringe. So this distance is the so this from dark fringe to bright fringe, the distance is basically the fringe width by 2. So this is the beta by 2, right? They are talking about what is the intensity at here. So intensity here is the maximum, okay. What is the intensity, maximum intensity? So guys, that is 4I0, right? 4I0. What is the minimum intensity? 0. They are asking what is the intensity in between. So it will not be okay, the maximum intensity they are saying is I0 only. So basically it is not 4I0, it is actually I0 if the we can take 4I0 when the intensity of individual sources is I0. So if this is also from one of the slit, we are getting I0, from other slit we are also getting I0. In that case the maximum will be 4I0, but here that maximum itself they are saying it is I0 only. Alright, so it means if we talk about the slit's intensity will be I0 by 4 and I0 by 4. Anyways, doesn't really matter here. So this is I0 intensity, this is 0. So guys, what is the relation between fringe width and this distance from the central maxima? So guys, we should know what is the relation between intensity and the phase distance, and from there we can find the path distance. So phase distance is intensity is equals to I max, okay, into cos square theta by 2. That is your relation between the intensity and the phase difference. So theta is the phase difference, okay. So if the phase difference is 90, so it will become cos square 45. If the phase difference is 180, it will become cos square 90. And we know so this is destructive interference, so the phase difference is 180. So here the theta is 180, okay, and here the theta is of course 0 because it is maxima. And as we go further, the theta, the phase increases linearly with the vertical distance. So here if theta is 0, here theta is 180, what is going to be theta here? Theta is going to be 90. So here the phase difference is 90. So if the phase difference is 90, can you find the intensity? Yes, very easily. Put theta as 90. So I will be I max, which is I0, and cos square 45. Cos square 45. So cos 45 is 1 by root 2. So 1 by root 2 whole square. So that is your I, which is going to be I0 by 2. So that is why the answer is B. So a little bit of complicated, but if you think very carefully, and one thing you should know is that the phase difference increases linearly with the vertical distance. So at beta by 2, the phase difference will be 90. Alright guys, let's continue. Okay, this is a little bit more conceptual, no calculation. So they are asking difference between interference and diffraction. So as we know, interference, there is only one source. The light from one source is coming and it is now going towards two different slits. Because if it is not one source, how can it be both are in the phase, you know? So both, it should be monochromatic source, and so that both the slit will initially both the slit will have the same phase. So that is why interference is always from single source. So interference is from single source, so that is why this is incorrect, because it is saying two isolated sources, which is wrong. B is saying from the same source, so that that should be correct, but let's look at the other one also. So no, in interference the wavefront is not same. There are two wavefronts that are interfering. So in wavefront, in interference the wavefront is not the same. So that is why this D is not correct. So sorry, C is not correct, because C is saying interference is from the same wavefront. So this is not correct, and of course D is not correct, because in diffraction the source is not same, it is the wavefront that is same. So you need to remember this one. Interference, there is only one source, light getting divided into two slit, okay, so two different wavefront. In diffraction it is only same wavefront which is getting diffracted. Alright, so that is the difference. Okay, next one. So what is the next one? Okay, yeah. Parallel beam of monochromatic wavelength is given, is normally narrow, width is given, focus convex lens. Okay, first minimum will be found for the angle of diffraction. So they are talking about angle of diffraction and minimum. So guys, the formula that we need to remember for this one is d sine theta, that is d is the fringe width, theta is the angle of diffraction, that is equals to m lambda, where m is the minimum, the first, second, third, whichever minimum. So here everything is given. This is of course 5000, this is 1, this is 5000 angstrom. Okay, this is sine theta is the unknown, and d is also given, which is 0.001 millimeter. So if you put the values, you should get the answer for the sine theta. All right. So yeah, not very difficult, I would say. Of course, you have to be careful with the unit, and that's all. You should be able to get it. All right, guys, let's continue. Okay, next one, unpolarized light. This is our polarization. So unpolarized light, intensity I is incident on two polarizer, angle between the polarizer axis is theta. What is the final? So guys, when light from I intensity comes, and let's say we have one polarizer, this is the one polarizer, light will go. So what will be the intensity after the first polarizer? So guys, polarizer basically it removes half of the intensity, because only one plane is allowed. So the light which is on the other plane, or basically the electric field amplitude becomes 1 by root 2 times, because if you take the ratio of electric field, so on one axis it will be E naught by root 2, in the other axis also on average it will be E naught by root 2. So the net electric field becomes 1 by root 2, and similarly the intensity will be half. So after, you need to remember this, after one polarizer the intensity becomes half. So this is I by 2 after one, but we have another one. So so when light goes from this, so guys, what is the relation? So guys, the relation is, if the intensity is I naught, if this is, let's say, I naught, so then after this one it should become I naught cos square theta, right? This is the intensity relation between two polarizer. If the angle is theta, then after the polarization it becomes cos square theta. So here initial is I by 2, so it will become I by 2 cos square theta. So that is your answer. Alright, so I by 2 cos square theta. So this is I by 2 cos square theta. Alright. Okay. Again, it is from, they are talking about what is the phase difference between these. So guys, we know whenever there is a medium, it creates a path difference. What is the path difference? So due to mu1, what is going to be the path difference? So the path of this light is mu1 into the L. Right, this is the path of this light, and what is the path length of this light? Mu2 into L. So we need to multiply refractive index with the actual length to get the actual path length. That will create the this will create the phase difference. So what is the path difference here? Mu2 minus mu1 L. This is the path difference. They are asking phase. So guys, phase difference, how can we find? So if the path difference is let's say lambda, one wavelength, what is the phase difference? 2 pi, right? For lambda it is 2 pi, for lambda by 2 it is pi. So for one let's say 1 meter of path difference, what is the phase difference? 2 pi by lambda. So for 1 meter of path difference, the phase difference is 2 pi by lambda. So for this, if this is the path difference, let's say this is the delta L, so what is going to be the phase difference? So basically it will be unitary method we are applying. So mu2 minus mu1 L into 2 pi by lambda. This is going to be the answer, which is option C. So unitary method for lambda it is 2 pi, so for this much it should be this into 2 pi by lambda. Okay, next one, thickness. So again guys, this is from path difference only. So mu1 minus 1 into the thickness of the plate, mu minus 1 into the d, which is, let's say t is the thickness, so mu minus 1 into t. So this is the path difference, and that is equals to n lambda, because it is creating a shift in the fringe pattern. So this is the formula we'll see. N is 4, 1.5, and t is also given. No, t is, they are asking lambda is given, 6000 Armstrong. So everything is given. If you put the value t, you can find easily, right? So yeah, if you do that, you'll get this is the answer. Alright guys, next one. Okay, this is from now. So optics is done, ray optics, wave optics. Now this is, we are talking about photoelectric effect, the stopping potential, all that. So what is this? Metallic surface is limited by wavelength lambda, stopping potential x. Same is limited by 2 lambda, stopping potential is x by 3. What is the threshold wavelength? Threshold wavelength, basically they are asking the wavelength at which the photoelectric effect will be there. So this is related to the work function. All right. So if I write the equation here, so guys, the equation here is hc by lambda, which is the energy provided, that is equals to work function. Some of the energy goes into the work function to eject the electron, plus then the remaining, this is the energy of the, which we gave. So some of that is lost in the electron emission, and remaining is basically the energy of the electron. Okay, so that energy of the electron is basically, to stop it, we apply stopping potential. So that energy is given by this term, E V naught. V naught is the stopping potential, right? So here now if we put the values, hc by lambda equals to work function plus initially stopping potential is x, so Ex. This is the first equation. Second equation is going to be hc by 2 lambda. So now the wavelength becomes twice, equals to W by E x by 3, right? Because it becomes x by 3. So if we subtract these two equations, we do this by this. So what is so they are asking what is the threshold wavelength, so at which we'll see. So if this stopping potential becomes 0, what should be the lambda? So what they are asking is this, hc by lambda dash equals to W. So this that lambda dash they are asking. So they are asking lambda dash, which is hc by W. So they are asking what is hc by W, right? So now if we subtract these two, so hc by we can first minus second we have to do. So hc by lambda minus hc by 2 lambda, that is hc by 2 lambda equals to Ex minus Ex by 3, so 2 by 3 Ex. So this is what you will get, right? This is the first, this is second. So what I have done is, this is basically first minus second, right? This is what you will get. So now what they are asking, hc by W. Okay, so this is the thing. So now we got the value of hc by lambda. So this we can cross multiply also. So hc by lambda is 4 by 3 Ex, right? So yeah, basically they are asking the threshold. So this, using this equation, this, let's say this is the third equation, and this one we can, so now we got the value of Ex, so we can find W also. Ex is what? hc by 3 hc by 4 lambda. So Ex is 3 hc by 4 lambda. So this I'll get 3 by 4. So W will be what? W will be hc by 4 lambda, right? From the third and first we can easily find W, which is going to be hc by 4 lambda. So now yeah, that will give us the answer, because they are asking hc by W. So hc by W is going to be 4 lambda. So that is why 4 lambda is the answer. So rest is just calculation. The most important equation is this one, that hc by lambda equals to the work function plus the this is stopping potential into the charge. So using that we can easily find. And what is threshold wavelength? Threshold wavelength means this lambda dash is hc by work function, where basically the stopping potential becomes 0. We are just able to eject the electrons. All right. So yeah, let's go to the next one. So again it is from photoelectric effect. So initial wavelength is given, energy is this. When wavelength is this, what is okay? So if this is 10,000 angstrom has this much energy, what is the energy of 5,000 angstrom? So when the wavelength become half, frequency is doubled, so energy is also doubled. So this energy, which is hc, this is hc by lambda. Let me write it here. So here the hc, okay, wrong color. Okay, hc by lambda for 5001, that is we have to double it, so 2.46 electron volt, that is given. Okay, now this whole saturation current, stopping potential are this much. What is the work function? So again guys, we have to follow this formula. that is we saw it here, hc by lambda is w plus ev naught. So hc by lambda is w plus ev naught. That is the formula. So hc by lambda is what, 2.46 equals to w, which is the they are asking, plus e v naught. So what is v naught here, which is 1.36 into 1.36. So now we can easily see work function. So this is 2.46 electron volt if I write the unit also. This is 2.46 electron volt. Okay, so 2.46 electron volt is W plus E into 1.3. So W is basically 2.46 minus 1.36 electron volt. So that is the answer. So what is that? That is 2.46 minus 1.36, so 1.1. So here actually this saturation current was not useful, because saturation current depends on intensity. It has nothing to do with the wavelength or the work function. Alright, so this was given just to confuse you, so this was not required. Okay, next one. Point source of light, distance 50 centimeter. Again guys, so this is from the, if we change the distance of the source, the intensity will change, but the frequency or the wavelength of the light will not change. So the stopping cutoff voltage, which is the stopping potential, it will not depend on the distance. So it will be same. That is why answer is same, because intensity is changing, frequency, wavelength are not changing. Okay, this one again guys, photoelectric effect. So anode potential. So here guys, as you can see, we have three different so what is this? So this, as you can see, these are the potential which are negative here, and at these potentials the photoelectric current is 0. Photoelectric current is on the y-axis, which is 0. So at these potential it is 0. Alright, so what we can say about these potentials, these is the stopping potential, right? This is the stopping potential. So in graph you can remember this one, that the potential which is where the graph cut the x-axis, that is your stopping potential. So which has the highest stopping potential? This one, nu3, then nu2, then nu1. So photo current is same, it means intensity is same, but if you talk about stopping potential, V3 has the highest stopping potential. So we know if the if I write the relation, so h nu is going to be work function plus the EV naught, which is the stopping potential. So if frequency is more, the stopping potential will be more, because this is constant work function. So if nu is more, V naught, which is stopping potential, will be more. So which has the most stopping potential? This third one, right, which is the most negative. So this V3 must be more. That is what we can infer. Stopping potential is more, so V3 must be more. So this is the maximum. Then we have nu2, then nu1. So V3, which is greater, and then nu2, which is more stopping potential than first. So that is the relation. Alright, wavelength of electron accelerated through potential of 1 volt. So guys, if we basically they are talking about what is the wavelength of the electron. So so this is what is the topic, de Broglie wavelength. That is where the wavelength of different bodies they are asking. So potential is 1 volt. If we talk about kinetic energy, which is here coming from the potential difference, which is EV naught, EV. So this is E into the 1 volt. Alright, that is your kinetic energy. So now we know kinetic energy. Can we find the momentum? Because the formula of wavelength is h by p, right? De Broglie wavelength. So h by p. So guys, what is p? Momentum. So can we write momentum in terms of kinetic energy? Yes, we can do that. So kinetic energy is what? P square by 2m. So p is going to be under root of 2mk. K is the kinetic energy, m is the mass. So this is 2mk. So if we put the values, root 2m, kinetic energy is what? E into the whatever is the potential here, EV. So now if you guys see h, Planck constant, m, mass of electron, E, charge on electron, so this whole thing is constant. Okay, so this constant thing divided by root of whatever the potential we are accelerating it. So this is the general formula. Okay, and the C, you can remember the value of C. Actually the value of C is this, 12.27 angstrom. So divided by root of V. So that is the general formula. You can calculate it also using calculator. If you solve this one, you will get this. So What is V here? V is 1. So that is why the answer we are getting is A, 12.27. If the V was, let's say, 4, so this will become half. Right? So this is the general formula you can remember directly. So if we accelerate electron through this potential, this is the wavelength of that electron. Alright? So yeah, let's continue. Okay, de Broglie wavelength increases by 25%, momentum decreases by P naught. What is the initial momentum? Again, simple. The formula we need to use is lambda equals to h by P, right? Very simple. So what they're saying is de Broglie wavelength increases by 25%. Okay, so this is, let's say, this is the initial lambda equals to h by P. Okay, that is what they are asking. Let's call it Pi. Now what happens? The wavelength become 25% more. So what is the wavelength now? 5 lambda by 4, right? Because it is increases by 25%, so it becomes 1.25, which is 5 by 4. So this is lambda by the new momentum. So let's call it Pf. Okay. Sorry, this is h, the Planck constant, of course. So this is the h. So now if I write Pf here, so Pf is going to be what? 4 by 5. So this is going to be 4 by 5 h by lambda, right? This is the final. And what is the initial momentum? H by lambda. So now if we calculate the difference, Pi minus Pf, because it is decreasing. So so it is, what is Pi? Pi is h by lambda minus 4h by 5 lambda. So this is be h by 5 lambda. So this is what this is given equals to P naught. This is P naught because the decrease is given P naught. So the h by 5 lambda is P naught, and what is, guys, Pi? Pi is h by lambda. So if h by 5 lambda is P naught, what is Pi? Pi is 5 times. So this is going to be 5 into P naught. So that is the answer, 5 P naught. So the only thing we used was the de Broglie wavelength, and rest will just follow. Just do the calculation and you'll get the answer. Okay, angular momentum of an electron energy is given. So guys, what is the formula of angular momentum? This we should remember directly, nh by 2 pi. That is one of the postulate of Bohr model. Angular momentum is nh by 2 pi. And we have to find only, you will get the answer. So energy is given. So guys, what is the formula of energy? It is minus 13.6 divided by n square. This is the value for the hydrogen atom. Otherwise, you have to multiply z square also, but hydrogen z is 1. That is the atomic number. So anyways, so this is what? Minus 3.4. So if we solve this, we'll get n square equals to 4, n equals to 2. We'll put n equals to 2, so 2h by 2 pi, so h by pi. That will be your answer. Very simple. Okay. Next one. So this is about energy level, wavelength of emission, all that. So this is also simple only, guys. What we need to do here is, this is from what is the energy of the wavelength that is comes out. So we need to remember the formula, and that is hc by lambda equals to some constant, okay, 1 by n1 square minus 1 by n2 square. That is the formula. So here and this is equal to the delta E, which is the change in energy. So from 2 to 1, energy change will be 4e by 3 minus E, that is E by 3, and from 3 to 1 it will be 2E minus E, that is E. So hc by lambda, this is for 3 to 1. So 3 to 1 delta E will be 2E minus E, so that is E. Okay, and for the other one now, hc by lambda dash, the delta E is 4E by 3, so that is E by 3. So you can easily find lambda dash from here. Lambda dash will be thrice of this, so that will be 3 lambda. Alright guys, magnetic field they are asking. Okay, revolving. So magnetic field, so if you remember the magnetic field formula, that is mu naught I by 2R. So I, we should remember that I is proportional to n raised to the power minus 3, and R is proportional to n square. This is from the Bohr model of atom. So this whole thing will be proportional to n raised to the power minus 5, right? Okay, transition with lowest frequency. So guys, this is again based on this only. So you can remember h nu or h frequency equals to delta E, or which is basically R 1 by n1 square minus 1 by n2 square. So this you can apply n1, n2, you can put different values and see for which it is the lowest. That is your answer. Okay, next one. X-rays, they are asking stopping potential. So guys, of course, stopping potential is more if the work function is less, right? That is the h nu equals to work function plus E V naught. V naught is the stopping potential. So V naught equals to h nu minus the work function divided by the charge on electron. So if the work function is more, the stopping potential will be less. So guys, in sodium actually the work function is less, and for copper the work function is more. So we can remember this one, W copper is greater than W sodium. So that is why work function for copper is more, stopping potential will be less. So for copper it is less, sodium it is more, stopping potential. Okay. X-rays, so guys, X-rays, what do they do? So they are rays, they have momentum, they will apply force also. So this is true. They will transfer energy, of course. X-rays have energy. When they collide, they will transfer energy. May cause emission, of course, photoelectric effect, they cause emission of electron. So yeah, the answer is all of these. Okay. Again guys, similar, same from this topic only, HF, which is, so if I write the formula here, so equals to R 1 by N1 square minus 1 by N2 square. So this is the formula. So electrons jump from third to second, and lambda, it is lambda naught, and now it will go from 4 to second. So one time we have to do, this is 3, this is 2, N1 is 3, N2 is 2 in the first case, and second case, N1 is, it goes from 4 to second, so N1 is 2 and N2 is 4. So when we can easily find the relation, very easy, based on this one. Okay, binding energy, let's talk about binding energy. So guys, binding energy is the energy which is extra. So how do we get the extra energy? So it is the, if we talk about the binding energy, that is mc square. So delta m, what is the, whatever the extra mass, into c square. So what is this delta m? How do we find this one? So delta m is basically the individual masses. So this is proton, which is mass of hydrogen nucleus is proton, neutron. So mass of proton, neutron are given, and nuclear mass of carbon is also given. So delta m we can find. Okay, how many protons are there? 6. So it is 6 into the mass of proton. Okay, mass of proton plus mass of neutron. Okay, this is the mass of individual minus the actual mass of the hydrogen atom, which is 12 AMU. So if you do this, you will get the delta m. Alright, so if you guys, if we put the values, this is a little bit of calculation. If you put the values, you'll get 0.099 AMU. This is delta m. And how do we find energy? So guys, it is this much. So, but there is a direct formula to convert delta m into the energy in mega electric volt, and that is binding energy. We can remember that is delta m into 931.4. This is the value in mega electric volt. So if you can remember, it is fine. Even if you forget, you can use this one. And yeah, so a delta m will be this. This is the value in AMU into this. So you'll get the answer directly from this. So if you know delta m, we can find the value directly in mega electron volt. Okay, next one. So this is a nuclear reaction. So X2Y alpha, guys, alpha is ejected. Alpha is what? Helium nucleus. So if I write the, let's say, atomic number of X, so let's say it is atomic number is A and mass number is Z. So now if we talk about Y, so guys, Y plus, so it is helium nucleus. So helium nucleus means 2 atomic number, mass number 4. So if we talk about Y, what is the atomic number? A minus 2, right? What is the mass number? Z minus 4. So of course X and Y are not related. So X and Y, so A and C are of course incorrect. We have to see B and D, which is correct. So now if we talk about the other reaction, so guys, Y, what is the mass number? A minus 2, mass number is Z minus 4. Okay, now nuclear reaction 2 beta. So beta is here electron. So if we talk about the Z, so electrons are ejected, it means the this A minus 2, the 2 electrons are ejected, so plus 2 electron. So it means the, if we talk about the charge here, so the charge must be A, because net charge is conserved in nuclear reaction. So this must be A, so that the A and this is minus 2, so total charge remains same. So mass atomic number will be A, and mass number of course will be same, Z minus 4. So now if we talk about X and Z, they have the same atomic number, same number of protons. So what are they called? They are called isotope. Same number of proton but different number of neutrons, so different mass number. So that is why X and Z, they are the same mass number, so they are the same atomic number, so they are isotopes. Okay, this is about half-life. This is about nuclear reaction. So guys, if you look at this one, what they are saying is from 1024 it becomes 128. So if we find the ratio n by n naught, so it becomes 128 by 1024, and that is what? That is 1 by 8. So it becomes 1 by 8. So in one half-life it becomes half, in another half-life it becomes half, and then another half. So 3 half-life. So it means this implies there have been 3 half-lives. And how much time is has passed? So 3 minutes. So in 3 minutes we have 3 half-lives, so it means half-life is 1 minute. So that is why the answer is 1 minute. Alright. Okay, next one, half-life. Okay, P half-life is 10 minutes and Q half-life is 15 minutes. So freshly prepared sample after 30 minutes. So 30 minutes basically means, for if you talk about P, so NP so for NP 3 half-life has passed. So NP will become 1 by 8th. So NP will become 1 by 8th N naught, right? Because 3 half-life. And what about Q? So only 2 half-life. So it will become 1 by 4, 1 by 2 into 1 by 2. So NQ in 2 half-life will become 1 by 4 N naught. So if you find the ratio NP by NQ, so that will be 0.5. NP is half of NQ. We can find the ratio and you'll get the answer NP by NQ. That is A. Alright. Okay, next one, N-type semiconductor. So guys, all kind of semiconductor are neutral only. The number total charge is 0, of course. So not just N-type, P-type also, the net charge is 0. Okay, interesting semiconductor, this much is given. So guys, we know the relation between the intrinsic semiconductor, the charge density, and when we dope it, so then the holes and the electron density. So guys, what is the relation? We need to remember this one. So Ni square is equals to Ne into Nh. Electron density, this is H is the hole density, Ni is the intrinsic density in the intrinsic semiconductor of the charge carrier. So here this is given 7.07 into 10 to the power 15. N they are asking the density of electrons, so Ne they are asking, and density of hole is also given, which is 5 into 10 to the power 22. So this of course you can solve and you'll get the value of Ne. You have to square this thing, both the side. Here you have to square, here on RHS it is fine. So you can find Ne easily. Alright. Okay, value of form factor in case of half wave rectifier. So guys, what is form factor? So form factor is basically, we need to know that it is I RMS divided by I average. Intensity, current RMS divided by current average. That is your form factor that we should remember. So RMS, if you talk about the RMS value, so that is I max divided by 2. Okay, and if you talk about average value, that is I max divided by the pi. So this will become pi by 2, which is 3.14 by 2, 1.57. So you can remember directly, half wave rectifier, the form factor is pi by 2, 1.57. Okay, this is about these. So in the diagram, the input terminal A and C, B and D, what is the output? So here you can see the diodes here, PN junction, and they are saying the input is between A and C. So if we talk about the input, so this is, let's say, some AC voltage which is there. B and C, the output, so the resistance is there between B and C. So what is the output and input related? So guys, if we talk about, let's say, the first half cycle, in which, let's say, this A is at higher potential. So if A is at higher potential, so this diode, let's say it is D1. So D1 is forward bias, right? Because A is at higher potential, so current will flow from A to B. But this one is the other, let's say, D2. D2 is not forward bias, it is backward bias. So from D2 there will be no current. So here there will be no current, right? So current will go from B to this point D. Now from D to C is forward bias. So let's call it D3. So current will flow from D to C. So the path of current will be like this. A to B, B to D, D to C, and then again A, right? So this is D1 and D3, they are forward bias. D2 and D4, they are backward bias in the first half cycle. Now if we talk about the other half cycle in which C is at the higher potential, so in that case D2, and actually this one, this is actually D3, let's call it D3, and this is D4. So in the second half cycle, it is the D2 and it is the D2 and D3 that are going to be the forward bias. So in the first half cycle, D1, D4 are forward bias, but in the next half cycle, the D3 and D2 are forward bias. So what is going to happen is the output will basically, it will be half of what in first half of the cycle we get the output, in second half we won't get the output. So that is why it is half. So in both the cycle there will be current that is flowing, right? In first half cycle the current will flowing through this, and in the next half cycle also the current will be flowing. So it is full wave rectified, because it's not that we are getting output in only first half, we are getting output in both the halves. So that is why it is full wave rectified, not half wave. If in one of the half wave, if the output was 0, then we can say it is half wave rectified, but here we are getting output in both the cycles. Okay, this one, so this is about the collector AC current for an alternating output. So guys, this is basically talking about the form, this is about the collector current, and here what is given, current gain is given. And so guys, we need to remember this formula, and that is the collector current is the current gain beta into the IB, which is the input current. Alright, so guys, beta is 50. What is IB? So IB we can easily find, so it is beta into V by R. Okay, so if we put the values, beta is 50. Okay, what is V here? V is 0.01 divided by R, which is 1000. So this is your answer. So if you solve this one, you'll get the answer 500 microampere. This is your collector current. Okay, next one. Here the emitter current is given. What is the change in collector current? So we need to remember this formula, guys. Again, collector current is the current gain, which is what, 0.98 into the emitter current. So change also will be similar. So this is actually change here. So doesn't matter even if it was actual value, we can use this one. So delta emitter current is what, 5 milliampere, and the gain is 0.98. So 0.98 into 5 milliampere. That will be your answer. So that is going to be 4.9. All right. Okay. Next one, logic gates. Okay. So this. How do we solve this one? So guys, the easiest thing is to make the truth table. So take this as 1, this as 1, and see what is happening. So 1 and 1, this is OR gate. So 1 and 1, the OR gate will make it 1, but this is actually NOR gate, so this makes it, so this becomes 0. So this is 0 and 0, this is again OR gate, so output is 0, but this is actually again NOR gate, so the output will be 1. So when it is 1 and 1, A and B both are 1, so output is also 1. So similarly, you can make the truth table. If this is 1, this is 0, let's say B, if you make 0, then what is going to be the output? So actually the output is going to be same. Okay, or let's see. So if this is 0, so 1 and 0, so that is going to be 1. So this is 0, 0, 0 again. So yeah, guys. So if you the thing is you have to draw the truth table. So give inputs to A and B and then see what is the output. And if you do that, you will find that it is a AND gate. So basically that is the idea. This, not just this question, but next one also. The idea is same. You have to draw the truth table, give the input of A and B, and then see what is the output, and then you will find it easily. So I hope you know how the gates work. So this is the, okay, this is actually AND gate. So 1 and 1, this was, alright, so basically, yeah, that is the idea. So this is the AND gate, right, and this is the OR gate. So if this is not, so this becomes a NOR gate, and this is AND, so this becomes a NAND gate. So NAND gate and NOR gate, these two are the things that are happening here. Alright, okay, final one, operational amplifier, guys. These are IC, integrated circuit operational amplifier, are digital IC. They are not logic gate. They are a combination of multiple logic gate and diodes, so and transistors. So these are digital IC. The digital IC and linear IC, the differences in digital IC, the input and output voltage, they have only two values, 1 and 0, low and high. In linear, the input output voltages, they can have, you know, continuous value. So in this operational amplifier, you have to remember this one, that it is a digital IC in which the input and output value can have only two value, which is 0 and 1, maximum and minimum. Alright guys, this is it. So this was the minor test 4, which is the later half of the 12th class, right? In 12th class, we did the second half from optics to the semiconductor. All that was covered here, and the first part was covered in the minor test 3, that was electrostatic till alternating current. Alright guys, so I hope this helped you, and not just this session, but giving the exams in general will really help you to help you prepare for NEET. And so be ready for mega test 1, which is the half syllabus. So of course the syllabus has increased, so you have to be more careful, you have to revise more quickly. Alright, okay, so yeah guys, let's end this one. Thank you for attending, and I wish you all the best for mega test 1.